Welcome to our bench to bedside session. I hope you all enjoyed this morning sessions. It is a great honor and pleasure to be moderating this session today. Aristotle University of Thessaloniki is my alma mater, so it's always a great pleasure for me to be here. Thank you for inviting us. We are very excited. We have a great a group of exceptional speakers today. They are all specialized in different fields of biomedical research. And each of them is going to show you how their research, how their work can benefit clinical outcomes and clinical care. We all know this, that bridging the gap between bed to bedside is really critical, essential to answering fundamental questions in medicine and molecular biology, from cancer field to neuroscience, to neurosurgery, to clinical trials. So we are going to kick off our session with an example of a very successful example of two scientists, two researchers, one physician from Mass General Hospital, Dr. Andreas Varkaris, and one molecular biologist, Dr. Ioannis Anidas from Roswell Cancer Center from New York University. That when they came together and they started working in this direction, how the research can benefit clinical outcomes for patients with cancer. So without further ado, let me introduce you to Dr. Andreas Varkaris, Assistant Professor of Medicine at Harvard Medical School, a hematologist and oncologist that works at Mass General Hospital and at the Temir Center for Investigational Therapeutics. Andrea, the floor is yours. Welcome.
So good afternoon from Boston. It's a pleasure to be with you today. I'm a medical graduate from Greece myself, and we would like to share with you how we experienced the first wave of precision oncology and how we try to guide the second wave of precision oncology to avoid mistakes that were done in the future and try to get better therapies for our patients. So I'm very happy to be with you today, and let's start. Can you see my screen? Okay, nice. Okay.
It's okay.
So let's start. So no relevant disclosures for me. I don't have access to my picture, so please let me know if anything happens or you lose connection. So I thought that it's a good idea to take you with me in a journey. 2010, I was a fresh medical graduate from the University of Athens. At that time, a brilliant scientist from Athens, and MD Anderson came back to Athens, and she tried to develop a GU clinic in the University of Athens. I was very fortunate to have her as my first mentor. And it was not difficult to understand that there were two major problems in oncology. First of all, the success rate of our treatments was very low. As you can see from this table, oncology does terrible, although it seems that we are the only ones who are reporting reliably the clinical outcomes of our trials compared to cardiologists. And the second one is time. So it took more than 10 years for a drug development, and that happened before 2016. Even with the reduction in timelines and the introduction of phase 2 within phase 1, or even in sometimes we start the phase 3 trial before even we finish phase 1 trial, time is long. And I want you to have in mind that most of the pharmaceutical companies that are running clinical trials have enough revenue for 6 months to a year. So you can understand why so many trials are failing, even in the case that the drug is working. So in 2011, we were in the midst of precision medicine and precision oncology. So we thought that by sequencing tumors, finding mutations, molecular alterations in the DNA of the tumors, we could match patients with available treatments and prolong their survival, even cure them. This slide comes from the NIH website, and I'm sure that most of the older scientists have seen it. At that time, Dr. Logothidis at MD Anderson Cancer Center was running a trial on a medication called dasatinib. It was a SARC inhibitor that was developed by Dr. Gallic and run by Bristol Myers. So the trial failed. So that was a disappointment for both of them. So they tried to create a program to avoid mistakes of the past and try to develop better microenvironment targeting agents for prostate cancer. So I was very fortunate to be one of their first postdocs in this program. And it wasn't difficult to understand why the project failed, because the two major components of the project, the two leaders, were so different from each other. The clinicians never understood the basic science, and the basic scientists never understood how clinicians think. And my job was to bridge the gap between the two worlds. So we started with a man on the left, on the right, Dr. Korn. He's currently the chair of GU at MD Anderson Cancer Center, and we tried to redesign clinical trials, and specifically phase 2 clinical trials. So the old approach, going from the lab to the clinic and testing drugs and molecular targets, changed. We started from the clinic, actually, because there was an ongoing phase 2 trial that Dr. Korn was running. The clinical trial was giving us responses and some molecular targets and some ideas about the response of the therapy and the human body to treatment. And then we went back to the lab and the animal hospital. We treated tumors from those patients with the same drug, and we analyzed the mechanism of resistance and the mechanism of action, actually, of the drug that, as you will see, was different than the one that was proposed. This was a big step for me, but a very small step for humanity, because by the time we published our results, it was too late. There were multiple ongoing trials targeting MET receptor in tumors that overexpress MET, including prostate cancer. Not surprisingly, during my residency and fellowship, I saw most of these trials published in journals that other than New England of Medicine, primarily because they didn't show improvement in survival of patients with prostate cancer or other tumors that overexpress MET. I was frustrated. Our results showed that MET was dispensable for prostate cancer growth. We showed that the drug worked through a different mechanism and will be more effective in more vascular tumors as kidney cancer. Fortunately, the drug is now approved for kidney cancer. But the main goal of my work was not successful. And it wasn't the only one. So the MATCH trial is the largest NIH funded trial in the US. Patients were screened and had next generation sequencing at baseline. And after that, they were allocated to different therapy arms based on the molecular profile of their tumor. The trial was run by Dr. Flaherty, and he is the person on the right. And I will take a break to say why I put pictures on my presentation. Well, while I was interviewing for my current job, I was interviewed by a person named Keith. Keith was Dr. Flaherty. I knew that Dr. Flaherty ran this trial, but didn't know how he looked like. And during my interview, I said that I disagree with the MATCH trial. So just have it in mind, try to put faces into names and don't repeat my mistake. And the MATCH trial actually is completing right now. We have publications from the trial. We have most of the arms completed. And it seems that the results are good, but they are not meeting the expectations. We believe that the reason that we find these results is because the decision for treatment arm allocation was based on laboratory findings, which are influenced by experimental conditions that we cannot meet in our patients in the clinic. You can never reproduce the experimental conditions that you created in a well or in a mouse in a patient. The patient have different comorbidities, different age, and different tolerability to therapy. Many times we are not able even to hit the target into patients. So looking as a junior, as a fellow, as a graduate fellow, I was looking for a job. And actually I was looking for a GU job in oncology and specifically prostate cancer until I met the person on the right, Dr. Yurik. He's a breast cancer oncologist and a phase 1 investigational therapeutic doctor. And at some point he told me, stop complaining, and I'm giving you the chance to have a real influence on drug development. So he provided me the appointment, the time, the support, and the place to really do what I wanted to do when I started with Dr. Korn, try to better understand targets in oncology and deliver them into selected patients with higher probability of response. It took us a few months to come to these questions. So we thought that during phase 1 development of the drug, we need to answer some fundamental questions that will guide the development of the drug and the class of the drug in the future. So the first question that we put is whether the target is relevant for cancer treatment. And I didn't want to do the same mistake with MET, because 5 years into my PhD, I found out that MET was not needed for prostate cancer growth. So I wanted this question to be first. Second one is whether the drug is a real drug. And we had to define what a drug means for us. And it's a substrate that circulates into the body, delivers to tissue, and has pharmacodynamic changes into the tumor. What is the mechanism of action of the drug? So we wanted to understand how the drug works early in the development and independently from the pharmaceutical studies. And then can we identify a biomarker of disease of interest and then guide future development of the drug class? And we were able actually to apply this to a relevant scenario. So most of you that have participated in an oncology clinic and have seen breast cancer patients, you have noticed that most of our patients these days take a CDK4 and 6 inhibitor early in their disease, if they have high-risk disease, or later after progression to hormonal therapy. These medications have actually showed a tremendous improvement in progression-free survival, and some of them have showed improvement in overall survival of patients with metastatic hormone receptor positive, HER2 negative breast cancer. The biggest question right now in oncology is what we do after progression to CDK4 and 6 inhibitors. And each oncologist is using his own strategy, and it's very difficult to support a specific one, but some oncologists change hormonal therapy. Some of them add a different CDK4/6 inhibitor. Some of them are adding a PI3K inhibitor in the case that the tumor is PI3K mutant. But there is no consistent answer to what we do after CDK4 and 6 inhibition. This is where it comes a phase 1 trial that targets a CDK2 inhibitor, CDK2 protein. This trial is sponsored by Pfizer, and this is a Pfizer developed medication. What we usually do in phase 1 trials, we dose escalate until we reach a dose that is tolerated by patients. And then we expand to get more patients into our trial and to combination therapies. Because of the promising results of this trial in the population of breast cancer patients that progress after CDK4 and 6 inhibition, we decided to create a translational research hub, and I will describe it later, around this trial based on CDK2 effect on a retinoblastoma. I looked for people around MGH that specialize in retinoblastoma. And one of the most famous labs in retinoblastoma therapy is the Dyson lab, and the most successful postdoc of that lab is Yannis. So that's why I emailed Yannis for his guidance on this project. And I was very surprised that Yannis immediately said, let's do it. I can be your co-investigator on this. And that's how our journey started. So this is how we imagine a translational research hub. So the Termier Center with a phase 1 development, and the patient treatment is in the center. Most of the members of the Termiar Center are physicians that have substantial experience in the lab. Most of them are, they have a PhD also, or spend more than 4 years in the lab. We have the molecular research lab that is attached to the Termiar Center, and this is the Sanida's lab, the Corcoran lab, and the Bulk lab in 3 different hospitals. We have the Bardisi lab that has expertise in model development, including mouse model and cell line development. We have Garis and Gardan, the 2 companies that are doing whole exome sequencing and circulating DNA sequencing, and the Lawrence lab that is analyzing the results. And we have the pathology at MGH and Roswell Park Cancer Center that is doing the staining for us and the functional analysis. More importantly, most of the members of our team can wear different hats. I usually use the picture of Messi on the right. I think all of us who, like those that know about soccer, know that he's not the best player. Zlatan Ibrahimovic is actually the best, but Messi was the only one who did this. So I give him a credit for that. Our research hub focused initially on the CDK2 inhibitor and the CDK2 inhibition in breast cancer patients. And we applied the questions that I described before into the specific example. Unfortunately, I won't be able to give you specific answers for the trial because this is an ongoing trial, but this is how the trial works. So each patient that enters our trial has imaging and molecular profiling at baseline. And when we say molecular profiling, we mean whole exome transcriptome sequencing. We check for target engagement, and we send for cell cycle markers. We have blood analysis for pharmacokinetics and also circulating DNA analysis that we perform monthly. We have tumor marker analysis to complement the clinical responses and the radiographic responses. We have model development with PDXs. We have not been able to develop a cell line from this group. And recently we added CRISPR screening through a pharmaceutical company that performs ex vivo CRISPR screening of the tumor cells. So this is how it looks. This is an example of the patient enrolled in the study. We have biopsies at diagnosis of metastasis. We have circulating DNA, tumor DNA, and tissue analysis. We have tissue at progression with analysis at progression. We have the duration of the patient into the study. And at progression to the study drug, we have a second biopsy and second analysis with circulating DNA and markers. We have matched responses with molecular profiling, and hopefully we'll have these results out soon. And this is how I want to finish my presentation with a translational research hub. I think that mistakes that were done in the past were related with delaying analysis of the target and the drug at early stages that we believe that we will overcome during this study. And closing, I would like to thank first of all our patients. Second, our donors. Most of our donors provide support after their treatment with cancer or a beloved person. And more importantly, our mentors. I think the mentor-mentee relationship it's one direction. We get from them much more than they give us. We give back. And I remember at some point, Dr. Logothidis told me that I'm giving you the chance to be one of the thousands of doctors to change cancer therapy. So we're grateful for them.
Thank you, Dr. Barkaris, for a fascinating presentation and for sharing your personal journey on research from bench to bedside. We're now going to move on to the next presentation, Dr. Ioannis Anidas, to complete the puzzle of the clinical trial that Dr. Barkaris was talking about. Dr. Sanidas is an assistant professor of oncology at Roswell Park Comprehensive Cancer Center at New York University. Dr. Sanidas, welcome. We cannot hear you. Can you please unmute your microphone?
Thank you. Hello. Can you hear me now?
Yes.
Great. And let's see if I can share my slides. Can you see also my slides?
Yes.
Okay, great. So I want to thank the organizing committee for inviting us to present our work in this conference that brings together Greek scientists from diaspora with Greek universities. So Andreas gave you a very nice description of what is the treatment strategy for estrogen receptor positive breast cancers in the clinic. And when Andreas came to me, asked some very simple questions. What is this? It's like most of the patients progress to CDK4/6 inhibition treatment. And the first question is, what is coming next? What do you suggest to treat this patient? And of course, because CDK4/6 inhibitors target cell cycle, it's obvious to ask if other drugs that target cell cycle can also show efficacy in the treatment of these patients. The third question was if it's possible to combine CDK inhibitors, CDK4/6 inhibitors with other cell cycle inhibitors. And of course, if we can work together to identify what is the mechanism behind this drug, how these drugs works, and of course, if we can identify biomarkers that can predict the response to the therapy. So I told him that to answer this question, we have to go back to the textbook model and see like how cell cycle work in the mammalian cell. I'm sure that most of you are familiar with this, but briefly to say that mitogenic signaling activates the kinase activity of cycling CDKs that phosphorylate RB. And this is the critical event, the inactivation of the retinoblastoma tumor suppressor protein for the cell to make the decision and start replication, initiate cell cycle. So the critical event for cell cycle progression in any cell line is the G1S checkpoint. And when the cell is able to inactivate RB and go beyond G1S checkpoint, there is no need anymore for mitogenic signals to maintain cell cycle progression. And this is the reason why cancer cells target the genes that regulate the G1S checkpoint. So in a cancer cell, we are losing this linear cell cycle model, and now cancer cells mutate the different components that can regulate G1S checkpoint to proceed to an uncontrolled proliferation. And as you can see here from this nice graph from a recent review, the mutations that occur in cell cycle components in cancer cells are mostly mutual exclusive. So cells, for example, that amplify cyclin E do not show mutations in cyclin D, or do not show changes, deletions or mutations in the retinoblastoma protein. So instead of having this linear model of the cell cycle, in the cancer cell, we have dependencies on specific genes that are mutated to deregulate G1S checkpoint. And going now back to the estrogen receptor positive breast cancer, what we see in these cells is that there is a transcriptional activation of cyclin D that enhances cyclin D CDK4/6 kinase activity. And now what is critical for these cells to enter cell cycle and initiate replication is only the cyclin D CDK4/6 activity. Other cyclins and kinases like cyclin CDK2 have suboptimal activity in these cells. And this is the reason why CDK inhibitors in combination with estrogen receptor inhibitors are so successful in the treatment of estrogen receptor positive breast cancer patients. However, most of the patients relapse to this therapy, and of course the question that is coming now is what is next, how we can treat these patients. And going a little bit deeper in the mechanism, the major difference in these breast cancer patients, as I referred before, is that the kinase activity that is essential for the G1S checkpoint in the cell cycle is the cyclin D CDK4/6. So when we treat these cells, this is a patient-derived cell line. Before treatment with any of the CDK inhibitor drugs, we see that although CDK4/6 inhibitors are capable to inhibit RB phosphorylation, and when we apply these drugs to these cell lines, we can see a very robust G1 arrest of the cells. That means that this drug can block cells before G1 checkpoint and stop cell proliferation. When we apply CDK2 inhibitor in these cells, paradoxically, we see that this drug does not inhibit RB phosphorylation. And as a result, we see that CDK2 inhibitors do not arrest cells in G1 phase. However, we see a G2M arrest that is indicated here with green color. This was a fascinating finding, and led us to ask the question, like, what are the differences between CDK4/6 and CDK2? And it is very well established from yeasts to humans that CDK2 has a non-redundant function relative to CDK4/6 in S-phase substrates, and is able to phosphorylate many of the DNA replication factors. So because of that, we built the hypothesis now that the suboptimal CDK2 activity that we have in estrogen receptor positive breast cancer is essential not for G1S checkpoint, but for the G2M checkpoint and the DNA replication in these cells. And this idea helped us to understand that probably it's not useful to target these cells with a combination of CDK4/6 and CDK2 inhibitors, because the two drugs target two different phases of the cell cycle. To make it more simple, it's like there is no reason to accumulate cells in G1 with a 4/6 inhibitor and try to kill them with a K2 inhibitor that targets DNA replication. These observations help us to understand a little bit better the CDK2 inhibitor monotherapy that is now in phase 1 at Mass General Hospital. And Andreas presented before a very nice overview of the study. To make it short here, just wanted to say that the beauty of this study and the capabilities of our group and our team is that give us the opportunity to collect samples from patients in different treatment stages. So we are able to have samples from the patient before going to CDK inhibitor treatment, from patients that are on CDK4/6 treatment, patients that progress to CDK4/6, so are the pre-CDK2 treatment, patients that are on CDK2 treatment, and patients that progress to CDK2 inhibitors. And from these clinical samples, we are able now to isolate cell lines, patient-derived cell lines, organoids, patient-derived xenograft models, and all of these models helped us to perform drug response assays, combination drug treatments directly to the patient-derived sample, and of course to perform in vitro mechanistic studies to understand better how the drug work individually in each patient. And in addition to this, we are able to analyze biopsies from these patients and perform whole exome sequencing analysis, expression profiles, multi-markers in immunohistochemistry or immunofluorescence sections, and of course, circulating DNA sequencing and collecting information, collaborating with bioinformatic groups and trying to identify biomarkers or expression signatures that correlate to the response or progression to the specific therapy. So here I'm going to give you a very small piece of data to show that our hypothesis that CDK2 inhibition targets G2M checkpoint is correct. And I show you here that CDK2 inhibitor shows an additive effect when it is combined with inhibitors of the G2M checkpoint. Specific here, it's a patient-derived cell line post-CDK4/6 treatment, and you can see from the synergy map that in specific concentrations of the drug combination, we have also a very strong synergy. In conclusion, we can say that CDK2 inhibitors in estrogen receptor positive breast cancer do not target G1S checkpoint, but targets DNA replication and G2M checkpoint, and this is an RB independent mechanism. Oh, yeah, this work is a huge collaboration between different cancer institutes, Roswell Park Cancer Institute in New York and Mass General Hospital in Boston. And many different labs contribute their expertise to bridge what we say bench with bedside. Just to say here that we are looking for people who are interested to work in translational research. We are offering postdoc positions, and if you are interested, please email me. Thank you so much.
Thank you, Dr. Sanidas. That was a very illuminating presentation. And it's a great example to see how molecular biologists can collaborate with physician scientists to benefit clinical trials. Thank you very much. So now we're staying in the cancer field, and we are moving to the next speaker, Dr. Anastasis Maniakas, Assistant Professor at the Department of Head and Neck Surgery at the University of Texas MD Anderson Cancer Center, to talk about cancer therapeutics on anaplastic thyroid cancer.
Hi, everyone. Can you hear me?
Yes.
Wonderful. Thank you. It's a great pleasure to be here amongst you all. I'd like to first thank Professor Turutoglu for the kind invitation, as well as the organizing committee. It's wonderful to see the wonderful work being done by colleagues, in other large cancer centers, and I'm very excited to show you some of the work that we have been doing. Yes, let me go ahead and share my screen. Let me know if you're seeing my slides. Are you able to see my slides, or?
Yes, yes, we can.
Okay, wonderful, great. So I'll quickly try to touch upon our experience, my experience in the effort to try and develop an infrastructure for the evaluation of novel anaplastic thyroid cancer therapeutics. As you mentioned, I'm coming to you from Houston, Texas, and the University of Texas MD Anderson Cancer Center. Let's see, moving. And I have nothing to disclose. So I'll try to quickly touch on these 5 learning objectives. I know I don't have much time, but I'll try to briefly touch on this to give you a glimpse of what we've done and what we're hoping to keep doing. So a quick update on anaplastic thyroid cancer. So thyroid cancers, as you know, come in all shapes and sizes, and thyroid cancers themselves will be also in different shapes and sizes. Most of the patients that actually have thyroid cancer don't even know they have it, or don't know yet, and they may have it for several years. Now, the annual incidence has actually increased in a dramatic fashion. We're talking about a 500% increase in the last 50 years. And it actually is the most common cancer in North Americans aged from 16 to 33 years old. It's also expected to be the 4th most common cancer in the United States by 2030. So there are different types of thyroid cancers out there. Most thyroid cancers, fortunately, are not as aggressive as anaplastic thyroid cancer. 80–90% of them will be in the more differentiated thyroid cancer that you see here, that are from follicular epithelial cells. You also have your medullary thyroid cancers that will be coming from your parafollicular C cells, and these are more associated to your multiple endocrine neoplasias. Anaplastic thyroid cancer only represents 1 to 2% of thyroid cancers itself. So why are we so interested in this? So even though it only represents 1 to 2% of thyroid cancers, it actually represents more than 50% of thyroid cancer associated deaths. And the historic 12-month survival for these patients is actually less than 30%, which is horrific if you think about it. And most patients will be presenting with very advanced disease. The most recent SEER database analysis actually showed that the median survival was 4 months, and less than approximately 1/3 of patients did not make it to 6 months. And this is this definitely puts it in the category of probably one of the most aggressive cancers in medicine. So how will a patient with anaplastic thyroid cancer usually present? Usually they'll be in their 60s. They'll have this rapidly growing neck mass. They'll have symptoms such as hoarseness, stridor, dyspnea, dysphagia. They'll definitely have lymph nodes that are involved almost always. And 1 in 2 patients will already have distant disease when they come to you. And if they don't, 3 out of 4 will likely develop distant metastasis within their first year. So it is no surprise that the AJCC has actually started the staging for this kind of cancer right away at stage 4, stage 4A, and then 4B, and then 4C when you have metastatic disease. So what can we do for these patients, and how can we make them survive longer? How can we treat them? How can we offer them something that makes sense? Because up until about 5 to 10 years ago, in most centers around the world, anaplastic thyroid cancer patients would be seen, diagnosed, and usually sent directly for palliative care or supportive care. So what are the latest survival outcomes at our institution, and what have we done in the last 10 years to try to change this? So in 2014, Dr. Cabanillas and Dr. Lai developed this program. It's called the FAST program, a catchy little name, but it really focuses on getting patients in quickly. So when a patient with anaplastic thyroid cancer comes in, as we know, time is of the essence. Usually when they come in, within 3 weeks, they can already be in severe respiratory distress and usually will end up passing away. So time is of the essence in getting everything done quickly. So first, we need to get them in to our institution quickly. We need to do significant workups, and this usually involves circular-free DNA analysis, staging scans, getting BRF-IHCs, and then also visiting a multidisciplinary team. And then we try to enroll most of these patients into clinical trials. Now, prior to this FAST program, it would take up to about 2 weeks to at least start seeing the patient from when we get contacted from a different institution. Now we've made it in less than half a day. We're able to set everything up. Patient comes in within 7 days, they've already had their workup and probably have already started some form of trial. So by doing this FAST program, first things first, we actually saw a 44% increase in the new anaplastic thyroid cancer patients coming to our institution. Being a very rare type of cancer, seeing 1 or 2 a year doesn't actually help you that much in trying to develop clinical trials or trying to create some form of advanced preclinical trials as well, as most of you will know. So increasing the number of patients that you see is the first key. And not only were we able to increase in the first year by 44%, after 5 years of running this FAST program, I was able to demonstrate that we actually increased by 200% in the amount of anaplastic thyroid cancer patients that we were seeing in our institution. So we really created a hub for this disease. And then it also increased the number of patients getting on clinical trials. But did this all increase or translate into an improved survival? And this data here that we published demonstrates a dramatic shift in survival in patients. So we went from what I had showed you at the very beginning, 4-month median survival, to 16 months. So we've quadrupled this in the span of 6 years. And the 6-month survival of the SEER data analysis was 35%, whereas at MD Anderson was at 80%, and 60% at 1 year. So these were dramatic changes for such an aggressive disease. Now, we obviously need to do more. And the only way to be able to really do more is to make sure that our preclinical trials are developing novel therapeutics. And this is now the next part of this talk, where I'll be able to demonstrate a bit how we've gone ahead and developed this infrastructure to use the tissue from these patients that come in with anaplastic thyroid cancer to develop PDX models and then use these models for our preclinical trials. So I'll try to simplify as much as I can the research model that was developed along with Dr. Lai, who was my mentor when I was training initially at MD Anderson, and now that I'm working in collaboration for a lot of the work. So it all starts with the patient and the tumor specimen itself. And if you remember at the beginning, when I said the FAST program, it's instrumental of getting these patients in. So once we get them in, we get the tissue, whether it's with a core biopsy, with an excisional biopsy, or for some reason they need an emergent airway. While we're in the operating room, I'll go ahead and remove some specimen. And this specimen is then used to run all kinds of analyses, whether it's biomarker evaluation, running next generation sequencing, running it to develop some cell lines, PDX development, or even tissue banking for future studies. In parallel, what we do is we try to screen for drugs, try to identify in an in vitro fashion potential agents or classes of agents that we'll be able to study on these developed cell lines or in these new developed PDX models. So once you have this along with your cell line and your PDX model, then you can actually run legitimate in vivo preclinical drug trials, as we did here. So a quick example, this will be, for example, a patient that I have done recently where we have a tissue specimen that's coming directly from the OR. I'll bring it down to the lab and then implant it into the flank of the mice. And what you hope is that within 3 or so weeks, all of these mice have developed these large tumors on the flank, and that all come from the same generation. Then what I'll do is I'll go ahead and remove this and then transplant it into future mice to be able to just multiply the generations and first of all show that we're able to continue growing this tumor and then on mice. And what you want is that when you actually take this tumor and cut through it, you want to see this necrosis, you want to see this granularity, the hemorrhaging, this kind of marble tan gray mucoid surface that is classically seen in anaplastic thyroid cancer. So grossly, it actually looks exactly like anaplastic thyroid cancer. But even on the microscopic level, you have this, the classic histologic evidence of necrosis, you have your spindle cells, you have the high mitoses. And when we demonstrate or show some of these slides from our PDX models compared to the tissue, from the human tissue itself, the slides, some of these are actually completely undistinguishable, which goes to show even more how good of a PDX model you can potentially get from this kind of cancer. In this slide, what I wanted to show you is just not even are we able to get good H&E slides that are comparable, but even our immunohistochemistry analyses are compatible between what we have on the patient and what PDX model, which goes to show that this model is valid. This model is something that can be used in trials, in preclinical trials, and hopefully give us ideas of how we can potentially treat these patients later on. So now we agree that the ultimate goal is to identify effective novel treatments while trying to minimize the patient burden with inadequate treatments, minimizing the cost and the time of a full-blown clinical trial. As my colleagues previously mentioned, I mean, a lot of these trials that are run by the pharmaceutical companies, or whether in collaboration with academic institutions, a lot of them won't even reach their endpoint, either because of costs, either because they've run out of funds, or because the drug just wasn't working. So why not test it on a preclinical reliable model? And one of the most reliable ones are these PDX models. So this is what we did here. We had done a high throughput screening where we identified 3 different drugs that we wanted to test that had shown interesting results on the cell line itself. Now, would this translate on the PDX model? That is the most important thing to first identify. So we used 3 different drugs: pralatrexate, LBH589, which is a HDAC inhibitor, and docetaxel. And what we saw is not only on the PDX model, but also on the cell line. I don't have the cell line figures here, but on the cell line and the PDX model and an orthotopic model. So we had injected thyroid cancer cell lines at the thyroid level of the mouse itself to kind of imitate the microenvironment. We saw that this drug specifically, which is an HDAC inhibitor, had demonstrated the best overall decrease in tumor growth. So this, in theory, becomes a drug that can then be tested on a phase 1 trial. Of course, toxicity levels need to be evaluated and all to be able to do that. But this gives you a clear example of how parallel analyses with a high throughput screen, identifying possible drugs, and then getting your PDX models ready to run trials, you can get this identification. And this, fortunately, this kind of preclinical trial, usually within 2 months, 3 months, you already have an idea of if you have a response or not to a drug. I'll quickly move into, I don't have much time left, but this last portion of our of some of the work that we're doing. So you identify the drug, you try, you test it. What is a even more efficient way potentially to see if it's working on a solid tumor or not? And one of the ways to do this, and a lot of research is being done in this now, is using the metabolic activity of a cancer. And a lot of the work that's being done from Dr. Lai and in association with Dr. Bankson on the imaging department is looking at how the metabolic activity of a tumor responds to treatment. And this, as we know, cancer metabolism is intimately associated with disease outcome. One way to measure this is through the lactate levels within a tumor. One way to do this, you can use hyperpolarized pyruvate that you inject in the bloodstream, and then one that's in there, you can then measure how the hyperpolarized pyruvate gets catabolized down to hyperpolarized lactate during by the tumor itself, and then you use hyperpolarized MRI technology to measure this activity. I'll give you a quick example. You'll see how this thyroid cancer. So you see here, you have a thyroid cancer before and after treatment. So this is a cancer that received a combination of pembrolizumab and lenvatinib, so an immunotherapy and a targeted therapy. And we see a dramatic decrease in the lactate production, correlating with a decrease in cancer cell activity and overall disease size. And what this tells us is pretty much in a real-time fashion, within 8 days of treatment, if we're going to actually expect a good response or not, as opposed to waiting a month or 2 months or 3 months to do additional PET scans or any sort of imaging. So here's another depiction of this patient's HPMRI results and kind of real-time evaluation of lactate activity production. And with this, very interesting findings, we're actually now running a trial where we're recruiting anaplastic thyroid cancer patients that are undergoing treatment, any type of treatment, depending on the type of clinical trial that they're on. And we evaluate the treatment response in a real-time fashion. And we really characterize the metabolic activity, and hopefully it allows us in the future to be able to predict in an earlier fashion if the patient or not will be able to respond to treatment. So if I quickly summarize the data that I have shown you, as I mentioned, the SEER data was quite bleak, 4-month median survival. And what we've already been able to do with all of this work is quadruple this median survival. And what we hope is that we'll be able to just keep striving and bringing this curve higher and higher in the coming months, in the coming years. And obviously, this kind of work is, as you all know, is done in a very multidisciplinary fashion. We have people that are involved in every possible facet of treating this disease, on the clinical side, the translational side, the lab side. And of course, this is a joint effort. And I'm very, very privileged to be able to be working with so many colleagues and so many mentors at the same time. Thank you very much for your attention, and I'm open to any questions.
Dr. Marniakas, thank you very much. That was a great presentation. Thank you. We are now going to switch gears and move into the neuroscience field. Our first speaker is Dr. Christos Lizgaras, Assistant Research Scientist at New York University Langone Health. Dr. Lizgaras is going to talk to us about a novel biomarker for Alzheimer's disease.
Thank you so much, Dr. Touroutoglu, for the kind introduction, and I would like to thank the organizers for the opportunity to present our work today. So today I'm going to be presenting about some new data that we have in the lab from animal models, and the data show a novel biomarker that we can record from the electroencephalogram. So the outline of my talk, I'm going to give you a brief background first, and then I'm going to talk about the overlap of epilepsy and Alzheimer's disease. And then I'm going to move on to show you some new data that we have. So the communication of cells in the brain rests on the generation of action potentials. These are spikes that are generated from the neurons, and those spikes are essential for the communication of the cells. All those spikes are organized in a framework that is called local field potential, that is contains all the information that the cells and the brain areas want to communicate with each other. So when we record brain activity from the brain, it looks like this. So it contains a lot of different frequencies that are important for memory and for our ability to sense the world and execute some functions. We can record the brain activity from the brain using a technique called electroencephalography. Here you can see recordings from the brain surface of a patient. We can do it in animals the same way, and we can get the information of what is happening in their brain, especially in animal models of disease. I don't know how many of you know that in Alzheimer's disease, we have signs of epilepsy, and those signs are actually seizures that are not manifested as in behavior, so they are pretty silent and we don't know that they are happening. Those seizures are at the early stages of the disease, and that is very important because if we treat them, we might be able to interfere with the progression of the disease. We can record these activities from animals that replicate many aspects of the disease, such as the pathology, amyloid plaques, but also behavioral deficits. So we took advantage of the animal model, and we recorded abnormal activity in their brains. And I'm going to give you an example of what I mean by seizures in patients with Alzheimer's disease. So here you can see the activity of the brain of a patient that was implanted with an electrode. That electrode is a foramen ovale electrode that give us the opportunity to record activities that are deep in the brain. So here you can see different traces. These are from the surface of the brain, so that is the scalp. And those activities here are recorded deep in the brain. So these activities we don't usually have access unless we penetrate into the brain and have access to those activities. As you can see here, there is nothing picked up from the surface of the brain, but when we record from inside the brain, there is a lot of abnormal activity. And this is quantified over here, where you can see that the foraminal valley electrodes pick up activities that are not seen on the scalp. So we have something that is going on that we are not aware if we don't record from deep inside the brain. So I would say that in Alzheimer's disease and in also many other diseases, we have things that we see, such as seizures that are clinically manifested, but also we have a lot of activities that we don't see, such as silent epileptiform activity in the form of interictal discharges. These are activities that are happening in epilepsy between seizures. And also we know quite a lot of things about high-frequency oscillations. So this is a pattern of electrical activity that is abnormal and is used in epilepsy to remove brain areas that generate seizures. So if we remove those areas based on high-frequency oscillations, most of the patients become seizure-free. So the question that we had in Alzheimer's disease is whether those high-frequency oscillations are also in Alzheimer's disease. To answer this question, we recorded from mice using a very high-density electrode array. So here you can see that we have 4 different electrodes, and each one of the electrode has 32 channels. So we can record from 128 spots in the brain. And here you can see an example of what we're talking about. We can record from many different areas. We can cover areas from the cortex throughout the hippocampus. So that gives us the ability to actually pick up some activities that are very focal, and otherwise we cannot record them. So here is what we recorded from our mice. So here is an example of a control mouse. You see, this is the activity that we usually get during sleep. It's very slow and it doesn't have weird components, let's say. But when we recorded activity from a transgenic mouse that models Alzheimer's disease, we could see something very atypical. So here with the asterisks, you can see some activities that are intermixed during the slow wave activity. And when we zoom more into the activity, we can see that something is happening that is not present in the control mice. And those activities are called high-frequency oscillations, and that they are abnormal. And they're happening very frequently during their sleep. So here you can see the quantification. We have approximately 50 events per minute. So when the animal is going to sleep and the human, we have some activities that actually disrupt normal sleep. And what was interesting is that in those mice, we recorded from 30 days of life up to 2 years of age. And we could see that those activities are independent of the pathology. What I mean by that is that at the late stages of the pathology, we have amyloid plaques. So here you can see a typical staining that we do in the lab to actually see whether the mice have amyloid pathology. And this is a picture from a 2-year-old mouse where you can see those green spots that are called amyloid plaques. Then we used another animal model that models the familiar cases of Alzheimer's disease. And in that case, the presenilin 2 gene is knocked out, and we found the same thing. We found that the animals had high-frequency oscillations during sleep. And those animals, they don't have amyloid pathology. So here you can see that their brains are clean, which means that those activities are independent of the pathology, meaning that if we can record them at the early stages and we treat them, we might be able to interfere with the disease. Interestingly, we had access to an animal model that models Down syndrome. Most of the patients with Down syndrome develop Alzheimer's disease after many years. And here you can see that the same thing is happening during sleep. High-frequency oscillations are interfering with the normal sleep architecture. So we quantify those phenomena at very different stages. So we quantify them when the animals were awake, but also at different stages of the sleep. And we found that those events were most prominent during sleep. And that is a stage of sleep when we consolidate memory and experiences. But also it's the stage of sleep when the beta amyloid is being cleared from the brain. So that means that we might have a functional link between how amyloid is being accumulated over time, depending on what is going on during sleep. And especially if we have abnormal activities like those, we might interfere with the normal cleanup of amyloid from the brain. As I said before, those activities are also present in epileptic brains. So here you can see some typical examples. So we wanted to actually do a head-to-head comparison between HFOs in epilepsy and in Alzheimer's disease. So as you can see here, those events are actually identical between the two diseases, which means that there might be some commonalities that we can take advantage of in terms of the mechanism that underlies their generation. Importantly, we could record those activities from the surface of the brain. So here you can see a typical example where we recorded inside the hippocampus, but also the overlaying cortex. And we could see that those events are broadcasted in the overlying cortex, which means that we might be able to record them with minimally invasive ways. And with that, I would like to conclude that high-frequency oscillations is a novel abnormality that is independent of the pathology. The commonalities that we found between Alzheimer's disease and epilepsy strengthen the idea of hyperexcitability in Alzheimer's disease, which means that we might be able to use anti-epileptic drugs at the early stages of the disease, and we might be able to interfere with disease progression. And the fact that we can record high-frequency oscillations from the scalp is a very promising finding because we might be able to record HFOs using non-invasive ways. And with that, I would like also to say that I'm a member of the Hellenic Scientific Association of the USA, and I would like to invite everyone to check our website and all of our programs that we are very excited about. And with that, I would like to thank you for your attention and enjoy the conference.
Thank you very much, Dr. Lisgaras. Very interesting and promising findings. We're moving on to the next speaker, Dr. Maria Stamou. Dr. Stamou is an endocrinologist and assistant professor in medicine at Harvard Medical School. She works at Mass General Hospital, and she will be talking to us today about a novel gene discovery in an inherited disease of neuroendocrine disorder. Thank you, Maria.
Thank you so much. Thank you to the organizing committee. Thank you, Dr. Torodoglou, for the invitation and the kind introduction. So since the theme of our workshop today is from bench to bedside, I would like to focus the discussion today just to go over from bedside to bench and then back to bedside. So as a physician scientist, whenever we see patients at clinic, to realize that there are some biological opportunities that we can take on and find the biology under the disease that we can help our patients with, and also their families. So for that, I'm going to be discussing today about the genetic architecture of rare inherited diseases, with misinheritability being a major challenge, and the role of non-coding variation, with a special focus in the disease model of isolated gene RH deficiency as an example of a rare inherited disease. So current screening strategies in all inherited diseases have helped identify the genetic background in about 50% of the cases. And most studies, either targeted or exome sequencing in clinical or research setting, focus on point mutations in the coding regions of the genes. And since 50% of the cases are considered solved, that means that the rest 50% are not solved. And this is what we call misinheritability. And this can be attributed to genetic variation that it's not captured by current screening tools. Those could be variants in the non-coding regions of the genome, either proximally to genes or more distally. Copy number variants, meaning large deletions, large duplications, complex chromosomal rearrangements, and common variations. And even though 98% of the human genome is non-coding, studies have only recently focused on that regions of the genome and have showed that it plays an important role in gene regulation. And so I would like to focus my talk today on the role of that non-coding region into the disease model of IHH, which stands for isolated hypogonadotropic hypogonadism. It's a rare disease caused by defects in secretion and action of GnRH, and our patients present with absent puberty and infertility. It's a rare disease, and even though more than 60 genes have been identified in the disease, 60% of the cases remain unknown, meaning that the disease is riddled by misinheritability, or in other words, in 40% of our cases, we do not know what the genetic cause is. And in addition, if we were to look at the normal distribution of pubertal timing, with early puberty being at the far left corner, delayed puberty at the far end of the spectrum, and IHH being at the furthest end, since there is absence of puberty, we have seen that genes that contribute to IHH actually also contribute to more common conditions. Though that can be the averages of puberty, menopausal timing, PCOS, and even cancer susceptibility. So just to make the point that discovering genes for a rare disease not only helps patients who suffer from that disease, but also can explain a lot of different common conditions that affect the majority of the population. And so just to give you an example about how we can go from bedside to bench and how non-coding variation may be playing a role, I'm going to be talking today about a case that was published actually more than 30 years ago in 1990 of a 22-year-old male who had presented with IHH, also had intellectual disability, type 2 diabetes, obesity. And at the time, he had undergone what was the state of art for genetic testing, which was a karyotype that had showed a balanced translocation, which arose de novo, meaning it wasn't inherited from any of the parents, and it was a translocation between chromosomes 7 and 12. And why the reason I got interested in this case was that traditionally we think that those balanced translocation, they're about exchanging genomic material between chromosomes without any gain or loss of genomic material. However, with whole genome sequencing, we now know that this is not entirely true. So how that works is that we take the human DNA, the genome, we shear it to small pieces, 3,000 base pairs, and then we sequence it and we map it to the reference genome. And if we see that one end, let's say, maps to chromosome 7, the other end maps to chromosome 12, we know exactly where the breakpoint is, and we know if there is any deletion, duplication of base pairs. And since I was very interested in the case, I decided to write to the first author of the paper, Dr. Best. I was not expecting any reply. That was from 1990. But surprisingly, he actually wrote back to me and shared that unfortunately the patient had passed away, but for some reason he had kept a blood sample and lymphocytes of the patient in an institute, and the patient's family was kind enough to give consent for our studies. So we got the sample and we did whole genome sequencing with the technique I just explained. We were able to find both breakpoints that were initially found in the karyotype, but this time we could tell exactly where the chromosomal breakpoint was. And it was interesting to see that underneath chromosome 12, there was a disruption of a novel gene. That's a non-coding gene. It's a low non-coding RNA called RMST. And when we look at almost 14,000 controls, we found that there was no such translocation, deletion, duplication at all, suggesting that it was a very unique case. In addition, we went back to a large cohort of IHH subjects. We already had exome sequencing on, but these gene is not covered by exome sequencing because it's in the non-coding region of the genome. So we did targeted sequencing of the gene. We found more rare variants. And actually, we found that common variants in this gene have been implicated in the age of menarche in a GWAS study. In addition, the common variation in this gene have also been implicated in intellectual disability, in diabetes, and obesity, all features that our patient presented with, implying that there could be an association between this single unique translocation and the presentation of the case of the patient. So in summary of the human genetic data, we, by studying a rare balanced chromosomal rearrangement, we identified a novel disruption of a non-coding RNA called RMST, and population studies actually showed that there is supporting evidence of its role in pubertal timing. So as we continued to learn about the biology of the gene, the case became really compelling. And the reason is that this gene is highly expressed in the hypothalamus, where the GnRH neurons are located, and it's critical for neuronal differentiation. So utilizing the patient's cells, and here I'm showing where exactly the breakpoint occurred in relation to the 3 transcripts of this RNA, we actually found that the patient's cells, shown here in gray, demonstrate reduced expression of RMST compared to controls iPS cells, suggesting that this translocation had a true effect on the levels of this RNA. And when in collaboration with Dr. Standon and Egg, we tried to differentiate those iPS cells to neurocrest cells, which is an early form of a GnRH neurons, we found, again shown in gray, that the patient cells are less capable of being differentiated into neurons, suggesting that this most likely is the cause of GnRH deficiency in this patient. Finally, we found that RMST is required for the function and is associated with proteins implicated in GnRH biology. It's required for binding of SOX2, a critical transcription factor for pituitary formation, for the hypothalamic formation, for neuronal differentiation. It also regulates known disease gene and actually regulates a known gene that has been recently implicated in early puberty. And it was interesting to see that we know when patients who carry mutations in McCorin-3, so they have reduced levels of McCorin-3 present with early puberty, whereas in neuronal stem cells, when we knock down RMST, we found an increase in McCorin-3, and we feel that this kind of like delays the puberty to the point that it causes absence of puberty and IHH. So just to say that RMST, it's part of a biological pathway, it interacts with known proteins for the disease, and it's kind of like a master regulator for this pathway. And this is what we actually found in the patient cells again. Again, those are the cells that carry the translocation with the low levels of RMST. They demonstrate low levels of SOX2, CHD7, but then increased levels of McCorin-3, confirming prior experiments. So in summary, we can definitely say that balanced chromosomal rearrangements are not that innocent that we thought they are, and they should be investigated further when found in patients' karyotypes. And we were able to uncover the first lone non-coding RNA in the disease, suggesting that the non-coding variants can play a role in this disease and other rare Mendelian diseases missing heritability. So in conclusion, we can all say that by studying the genome, trying to identify single nucleotide variants, complex variants, deletions, duplications in coding and non-coding regions of the genome, both rare and common, can help us close the gap that exists in this missing heritability and in the genetic architecture of IHH and other diseases. And I believe this case kind of highlights the way that when we see patients at clinic, by performing deep phenotyping, genetic analysis, we can identify biological pathways and a network of proteins that can then be targets for both diagnosis and therapy, not only in this disease, but other conditions that may be influenced or governed by the same biological network. And with that, I would like to thank everyone for listening. A special thanks to our participants, the families, and the HBF USA. I can say I'm really excited and honored to be a board member, and we can take questions at the end. Thank you very much.
Thank you very much, Dr. Stamu. Really interesting findings, and looking forward to see what's next. we are moving from neuroscience to neurosurgery. We have a renowned neurosurgeon today with us, Dr. Nicholas Theodore, professor of neurosurgery at Johns Hopkins University. Dr. Theodore, welcome. We can't hear you. Could you please unmute your phone?
How many neurosurgeons does it take to unmute the phone? Can you hear me now?
Yes, we can.
All right. Well, thank you very. It's really a great source of pride. And can you see my screen?
We can see your screen now. Yes.
Okay. Well, I wanted, first of all, I'm honored to be a part of this, and it is with absolute great pride. Obviously, as a Greek neurosurgeon, my grandparents came to the United States from Greece at the turn of the century, but my wife's parents were born there, and obviously this Greek culture and my heritage is really the most important to me. We're going to change topics here a little bit, and we're going to talk about some advances in spine surgery and things that I've been working on for the past 20 years. These are my disclosures, some of which are relevant to this talk. But if we go back into antiquity, we see here the figure on the right was the god Hephaestus, and really has been an inspirational figure to me. Hephaestus was the Greek god of metalworking, and he was also the god of technology, and really the one who Zeus called upon to design and develop new technologies. When Zeus had a horrible headache that was unrelenting, that was going on for days, he summoned Zeus to help him, and Zeus actually performed the first craniotomy and opened up Zeus's head, from which obviously his daughter Athena was born. Those of you who have daughters might understand that that could be a potential problem. But the reality is that, in antiquity, that was really the first description of craniotomy in ever. And I think that it is fitting that neurosurgery has become a technologically advanced specialty, and the interplay, dating back to antiquity is certainly one that should always bear discussion. So I'm coming from you today from Johns Hopkins, and really has also been a center of neurosurgery in the United States. And where I am sitting right now, where that yellow stripe is the Carnegie Center. The Carnegie Center was the old operating room for 100 years here in Baltimore, where neurosurgery was done by Harvey Cushing and Walter Dandy. And now that same space is no longer an operating room, but has become a technology center. And it is from there that we have made significant advances in our field. And those include robotics and other things we'll talk about briefly. This is the way I was taught to do surgery. We had to open the patient. We see the thecal sac in the middle, and we're palpating here to be able to put a screw in the spine. And that, fortunately, is going by the wayside. So opening up patients like this, we're not doing anymore. What changed our field radically was the ability to use image guidance. And this was back in 1987. And, shortly thereafter, I adopted the ability to use real-time image guidance to look to see where we were in the spine, to be able to guide us without having to make a big incision. The problem with that is, as you can see here, I'm looking away. Our attention was always deflected. So the question came to me was, are we able to get to the location we want to reliably, quickly, with the use of a robot? And that concept came to me 20 years ago while working in Phoenix, and was able to conceive of and design something that would allow us to navigate in real time, to be able to do spine surgery without using our hand and eye to look back and forth at a monitor. And this was our first iteration of the robot. This is about 10 years ago. The first time ever, this is in a cadaveric specimen. We're cannulating here through the robotic arm. And then what the aha moment was that we could do this. We could actually, through the skin, put in hardware very accurately utilizing a robot. Several years later, we were finally able to bring this through the FDA, and I was proud to say that the very first robot in Europe was in Greece, in Athens, and they're still using that currently. But just to show you what this is like, we're able to now plan where we want the instrumentation to go. So on the screen here, you see The screws are planned, for instance, if we're putting in instrumentation in the spine. And instead of making incisions, we make a marking pen where we want the incisions to go. We're able to make very small incisions. And then with those small incisions, then utilizing the robotic arm to drill and really percutaneously put instrumentation in. This is really going to change the way we do things. Our blood loss has gone down, the procedures become much quicker, and you can see here in surgery, we're watching in real time the robotic arm situated over the patient and the instrumentation going in. This has really been a disruptive technology in our field and one that will change things. I think when we look at the adoption of robotics in surgery, this is increasing on a daily basis. And some of the things that we're doing, this is a case from just a year or so ago of a woman with a horrible spinal deformity and osteomyelitis. These are very difficult cases with deformity because the anatomy is distorted, but utilizing robotics and image guidance, we're able to very quickly place her instrumentation and very accurately reconstruct the spine. This is a trauma case in a patient who weighs almost 400 pounds. Difficult, but again, what you see here are very small incisions, and with the usage of robotics, in about 1 hour, we're able to do this case. We're also using this technology in the cervical spine, which had never been done before, to repair fractures. So this is a Hangman's fracture here, you see, and ultimately with some spinal cord compression. But utilizing the robot, we're able to literally repair that fracture without having to fuse across the atlantoaxial joint. Again, changing the way we do things. This has improved our accuracy, and what we're seeing now and publishing on extensively is the fact that while using our hand is accurate, our misplaced screw rate is about 10%. So 10% of the time when I use my hand alone, the screw doesn't go exactly where I want. With the robot, we're in the 99 percentile range. And again, where our accuracy is increasing. This is from the lab just about a week ago. The next step is going to be not just putting hardware in, but being able to drill bone and cut the bone. So this is a scalpel that's been designed to cut through bone. And we're now looking at how we can adopt that with robotics. I'm going to change topics here a little bit and talk about spinal cord injury. And this is another project that we're working on heavily that's funded by the Department of Defense. And one of the issues with spinal cord injury is that we don't know what happens. After somebody has a spinal cord injury, we have no way to effectively measure perfusion of the spinal cord in real time. So what we're looking at is the ability to, in somebody who's just had a spinal cord injury, to place a sensor on the spinal cord to allow us to monitor blood flow in real time. This has never been done. So we conceived of this idea of utilizing ultrasound to give us basically an idea of perfusion at the site of injury so that we know how the patient is doing, how we can treat that patient. So we see that we in the lab started looking at ultrasound. These are some pictures of what the resolution could be with high density ultrasound. You're looking at actually the blood flow in the spinal cord in real time. This has never been done before in large animal or in humans, and we're in the process of developing this technology now. What I will say is that we have built the patch, and hopefully in the next year, we'll be able to start human clinical trials where this patch will be implanted at the time of injury to allow us to monitor blood flow in the ICU for the days following injury so that we understand when autoregulation comes back and when patients are getting better. This will be done in conjunction with this device here, which is going to be a catheter that we place that allows us to drain fluid and also measure biomarkers in real time. And the last thing I wanted to talk about is some other exciting technology in spinal cord injury as well, which is scaffolding. Anytime there's a spinal cord injury, we can see here normally what happens in about 12 weeks is that we get a cavitation of the spine. So the idea was, can we build something to fill that gap? Can we put a scaffold in the spinal cord? And if you think of the scaffold, putting it in the spinal cord, think of it as a three-dimensional bandage, which allows the spinal cord tissue to hang on to and grow through to promote healing after injury. So on the left, what you see is a control animal that has had a significant spinal cord injury. And on the right, what you can see is the remnants of the scaffolding in the middle. But look how much more tissue is preserved once we open the spinal cord and place that scaffolding. And we can actually see and have demonstrated tissue growth through the scaffold, which could promote healing after spinal cord injury. So this was the first patient done a couple of years ago. You see this horrible spinal cord injury, and this was done in 2014. The trial still goes on. What I'm going to show you here is the spinal cord being opened, and this is the individual who had a spinal cord injury just several hours ago. And we're measuring here is a very small corridor here, and then this is the scaffold actually going into the spinal cord. So the scaffold goes in, and then we wait. And this is about a month after surgery. This patient who came in without any ability to move his legs at all is now moving his legs. It may not be the scaffold. It may be opening up the dura. It may be opening up the spinal cord, but this is something that holds great promise. We've now done 21 patients and published our series and hope to continue in that regard. Imagine, though, that scaffold may at some point be impregnated with stem cells, for instance, which may allow growth. So this is a future for spinal cord injury, which I think is getting brighter every day. Ultimately, our field is very technologically driven, and I think that dates back to the time of Hephaestus. But I just wanted to take this opportunity to thank the organizers of this meeting for allowing me to participate. And again, it's with great pride that I'm here today. And next year, I'm coming in person. So thank you very much for your time and attention.
Thank you, Dr. Theodore. Thank you very much for a great presentation showing us how research in engineer can really help shape the future for spinal surgery. Thank you very much.
My pleasure.
Now we are moving connecting with Australia, and we have a great speaker, Dr. Vaso Apostolopoulos, a Pro-Vice Chancellor, Research Partnership, Victoria University in Melbourne. She's going to be talking with us about what we have learned in the past 25 years on research from bench to bedside. Dr. Apostolopoulos, welcome.
Thank you, and welcome from a very cold Melbourne late night. I'm gonna. Can you see my slides?
Yes, we can.
You can see. Okay, so I'm going to give you a bit of a summary of what we've learned in the last 25 years from going from bench to bedside and back to the bench, back to the bedside. And we've been going backwards and forwards for the last 25 to 30 years. But what have we learned in doing this? And in particular, I'm going to focus on some of the work that I've done on developing vaccines for cancer. I've done a lot of other work on drug development, vaccines for other diseases, etc. But I'm just going to focus on this aspect of my research. So I'm going to take you back to the first paper I ever published, which was in 1993. I did this work in 1991. It was part of my honors degree. And what I did back then was I made monoclonal antibodies, which basically our publication, we published a paper describing the production of monoclonal antibodies. Now, these days, these antibodies are sold by various companies that sell antibodies, and it's just one little antibody inside a methods paper or inside a paper that people publish. It's just one antibody used. But back then, 1993, 30 years ago, we were able to publish a whole paper on development of monoclonal antibodies. But what was important about developing monoclonal antibodies was we were able to, I generated these antibodies, and we were able to pick up at the time some cancer proteins or antigens that were overexpressed. Here is a breast cancer, the immunoperoxidase stain, and the brown staining shows positive for this protein, which we called MUC1, mucin, which is overexpressed on cancer cells. It's overexpressed on adenocarcinomas, so little expression on the normal breast tissue. And at the time, it was actually quite exciting. We did a lot of work on trying to identify cancer markers, and we had developed early diagnostic kits, which were marketed and used for serum tests, etc. But it was a marker that was found that was overexpressed on the cancer. So what we did was, to cut a long story short, we did some clinical trials thinking that if we could target an immune response to this antigen or to this protein that's expressed on the cancer, will hopefully stop the cancer from growing. So we did a phase 1 clinical trial. We published it in 1995. The work was done 1 or 2 years prior to that. And it was one of the first clinical studies where we used this antigen as a target to develop vaccines. What we found at the time, we used peptides from this protein that's overexpressed in the cancer cells, and we actually conjugated to a common carrier diphtheria toxoid or tetanus toxoid. We had conjugated in an attempt to try and generate an immune response. What we found at the time, we found that this method was safe. It generated antibodies. There were some T-cell responses. We really were more interested in the T-cell responses. And there was very weak clinical responses. But what we learned from this study was that this actually opened up and paved the way for translational research. So, so we can actually target proteins on cancer cells and try and generate immune responses. So from this, we found that, there weren't really any strong clinical responses. We were able to stimulate an immune response, which was, as an immunologist, we were excited that we were able to do that. So we went back to the lab and we thought of, okay, how do we improve on this vaccine delivery system? How do we generate stronger, more powerful immune responses? So I did quite a lot of work, and this cell here became my best friend for quite a long time. It's called a dendritic cell. So these are the cells that actually you need to target or deliver your vaccine or your antigen, because these are the cells that go and educate these killer T cells. Okay, so I had to come up with a way of delivering this antigen that we found was expressed on the cancer cells. How do we deliver it to these dendritic cells? And then in turn, how do we find a way of them stimulating these T cells? Lots of work was done in mice, rats, rabbits, chickens, rhesus macaques, lots of publications way back then. And then we did some clinical trials. This is now a targeted approach. So we improved. So we previously we had just the peptides, but we showed that they were able to stimulate some sort of response. Now we were able to improve it, where we're targeting it to these antigen presenting cells, to these dendritic cells, to get a better and a more enhanced immune response. Clinical trials were conducted, a range of clinical trials were conducted. It wasn't just one, but we did this one here was published in 1997. So we're going way back. So I'm doing a little bit of a summary of what we've done and what we've learned. A phase 1 clinical trial, obviously, we did a dose escalation. That's this paper here on the left. We also did a phase 1/2 where we did the route of immunization, subcutaneous. There was another study. Participants had intramuscular injection, intradermal injection, and even intraperitoneal. That was a colon cancer where they put a catheter within the peritoneum and will inject the vaccine in there. And that was published in the year 2000. In the year 2001, we came up with a new way of analyzing immune responses. So we did all these cytokine analysis and flow cytometry. So it was actually a big advance in how to measure responses. So you do a clinical trial. At the time, we wanted to find out whether we're generating responses, but how do you measure these responses? So we did a lot of cytokine analysis, which was new at the time. These days, it's straightforward, but at the time it was quite complex. what we found that, you know, we needed to give 4 injections of the vaccine. Even after 8 injections, antibody responses, we were able to increase the antibody responses, but that's 8 injections. But taking note, this is a self-antigen which is overexpressed on the cancer. So we're trying to break tolerance in a way. So it's actually, you need quite a lot of injections. But importantly, what we found that cytotoxic T cell responses, so T cells were generated in some of these patients. So, I mean, just to do these phase 1/2, another one, this one, another clinical study. I mean, these took, you know, about 4 to 5, 6 years actually to complete all these studies. Okay. We then went on and did a pilot phase 3 clinical trial. So it was in early breast cancer patients, and we injected them with the vaccine. And there was a double-blind placebo-controlled study. So half got the vaccine, half got a placebo. They were all on tamoxifen, which was a standard treatment for this cohort of participants. And this started in 1997. There was a 8.5-year follow-up was the paper on the left. We published that in the year 2006. 15-year follow-up data, we published that in 2013. 18-year follow-up data, that was published a few years after. We now have a 24-year follow-up data. So, you know, we the whole world has heard about the COVID vaccines, and people are jumping up and down saying, what's going to happen, you know, with the vaccines long term? Are there going to be any side effects, etc.? But with what we have done here is we've actually followed up these patients, or the clinicians have. They've followed up the patients for 24 years. We've got that data, and we're looking to write that up for a letter to the Lancet, so just to show that 24 years later, they are all protected against recurrence, compared to the placebo, which 24 years later, there's about 60% recurrences in those patients. So as a protection, we were able to show that we could get protection against recurrent disease. In the meantime, we were back in the lab, and we were trying to further enhance the vaccines, so try and get even stronger immune responses. So we came up, this was around the towards the end of the 1990s. In the year 2000, we did the first clinical trial where we isolated monocytes, leukapheresis from patients, monocytes. We cultured them in labs. In the labs, from the monocytes, we actually added some cytokines, and were able to generate these dendritic cells. Okay, so this is monocyte-derived dendritic cells, and then we mixed it with the vaccine overnight. And then, so the dendritic cells mixed with the vaccine, and then you re-inject. This whole process takes about 6 days. You need special clean rooms, GMP facilities, PC3 labs. It's quite complex, very tedious, very expensive process, but it's to take out, you know, so we take out the cells, we generate the dendritic cells, mix with the vaccine, and re-inject. So this was done early on. You know, these methods are common these days. But what we did, this was done in ovarian cancer patients, because there weren't many treatments for ovarian cancer patients. So what happened was, before the trial, and then these are some scans that the clinicians have given us, and this was 14.5 months after treatment. There was some lung metastases which were no longer present, and some liver metastases. So a range of clinical trials were done, phase 1, a phase 1/2, a phase 2B, but all these took about 10 years to do. It was because it's the funding that you needed. The funding, we found an industry partner, etc. So these things take a long time. Although this process was quite, we also saw the cancer marker CA125, which is a cancer marker for ovarian cancer. Once they were getting the vaccine, the cancer markers also went down. So although it was very interesting, we got really good responses. It's actually not for worldwide use. I mean, you need these sort of facilities. These facilities are not everywhere. I mean, most countries don't even have these facilities. In Australia, we only have 2 or 3 labs, facilities that are able to do something like this. So, you cannot actually do this treatment, take the vaccine and go and inject anybody anywhere in the world. So, one, it's not applicable, it's not appropriate or convenient for worldwide use, and it's also very expensive. It's about $80,000 per patient to do this. So results were really good. It's called CVAC. If you want to learn more about it, that's a vaccine that we developed for ovarian cancer. But what we did, we're back in the lab to try and improve on coming up with other ways of developing vaccines. So, I'm not, this is just a summary. There's 500 publications that we published on over the last 30 years. But, we've done a lot of work on improving vaccines. I've made some DNA vaccines, peptide-based vaccines, vaccines with nanoparticles, using a range of different antigens, lots of adjuvants, etc. We've also designed vaccines for other disorders, autoimmune disorders, infectious diseases. We've made a few vaccines for COVID, etc. Here is one of our MS vaccines that was on the cover of one of the journals. And we're currently seeking some funds to go into human clinical trials for some of these vaccines. So what have we done in more recent years? Because of technologies have advanced, what we knew back in 1993 when I published that first paper, that was one antigen, one protein that was expressed on the cancer, and that's all we knew. We didn't really know much about the microenvironment, the cancer microenvironment. We didn't know much about what does the cancer do. It has a lot of immunosuppressive molecules. So these days we have learned a lot more about cancer. There are a lot of cells that are involved in the microenvironment that contribute, and you could stimulate and could have an effect. These are all different types of immune cells, and also there are a lot of immune suppressants. And one of the popular ones is PD-L1. PD-1, that's just that one there. But there's a lot of other immune suppression markers and things that happen that the cancer suppresses the environment. So a lot of these we've done a lot of work in these areas. We've published lots of papers in these areas. I won't go through it. But from here, we actually have learned more about the cancer environment, how the cancer behaves when there's the immune cells present, and from there we can actually improve upon vaccines and also in drug development. I'm not sure what that's there, but also with the recent next generation sequencing, we've done a lot of work where we've sequenced a lot of the cancer cells, and with various treatments that we use for some cancer cells, and this is important. I'm not sure what's happened here. Anyway, so with one of our treatments, we actually have found vitamin B6. We actually have found that it actually stops cancer cells from dividing. We weren't sure why it was stopping cells from proliferating. What we did was we did this next generation sequencing, and we found that it actually is involved in this. It downregulates the mevalonate pathway, which degrades this mutant P53. So it restores the wild P53, and therefore it stops cancer cells from proliferating and dividing, and get cell death, etc. So there's something as simple as environment, but we've come up with a lot of different other drugs or mechanisms. So basically what I'm trying to say here is these new technologies that have come up in the last few years, even next generation sequencing, is actually opening up a lot of pathways and information about how to manage and treat cancers. And from that, you could even develop more improved cancer treatments, vaccines, drugs, etc. This is a paper that we've just got accepted where we actually got, we had 300 samples from one of my collaborators in Greece. He's a breast cancer clinician, and he had breast cancer tissues from a range of different patients from stage 1, stage 2, stage 3, stage 4 cancers. And what we did is we looked at single nucleotides, SNPs, and we actually found that there is polymorphisms, interferon gamma gene polymorphisms in these Greek breast cancer patient tissues. So that it opens up new knowledge and new ways of trying to treat and understand cancer and how to treat it. Furthermore, there's a lot of work that we've been doing on, and what we have, people around the world too, where new markers on cancer cells and new proteins. So, back 30 years ago, we had that one protein that a lot of work was done, but in fact, there's not just that one. There are endless amounts of markers and proteins that are expressed on cancer cells, which we have learned over the last 25 years, that there are multiple antigens and proteins expressed on cancer cells. So the idea now is to make a vaccine using a multiple antigen approach. So instead of targeting just that one protein on the cancer, we can now target many cancers. And this is actually one of the colon cancer. It was one of my students' work that was on the cover of this journal. So to sum up, we are using new knowledge to develop new improved vaccines for cancer with new delivery systems. One antigen is not enough. We have to use the multiple antigen approach, because now these multiple proteins have been identified, so we could use a multi-targeted approach and stimulate immune responses, antibodies, T cells against multiple antigens. There's a lot that we've learned from the cancer microenvironment, and we are still learning a lot. There's a lot of immune suppressive markers. We've also found that there's interaction between the neurological system, the immune system, and the cancer. So there's a crosstalk there. And so a lot of this information puts in a lot of puts a lot of pieces to the puzzle, and we could improve on our vaccines. NGS, bioinformatics, single cell analysis, single nucleotide analysis, a lot of this work that have been done further increase our knowledge, and we could improve on developing vaccines. And yeah, we're actually working on a lot of other diseases. We're working on autoimmunity, type 1 diabetes, MS, rheumatoid arthritis, infectious diseases, COVID, a lot of other infectious diseases, a lot of rare diseases, tropical diseases, neurological diseases, etc., on developing vaccines and drug development. So this is my final slide. I think immunotherapy, what have we learned over the years is that, there's a lot of information that we're gathering now that we did not know 30 years ago when I first made these vaccines. Although we did see some clinical responses, we did see prevention of recurrent disease in early stage breast cancer patients. However, we can further improve on that from what we've learned over the last 30 years because actually cancer is a lot more complex than what we thought it was. But actually, what have we learned from COVID? This is not a COVID talk, but what have we learned from that? We actually have learned a lot, because we're able to see vaccines developed at a very fast rate. I guess the reason why there was such relatively fast rate, I've been working on vaccines for 25, 30 years almost, and we're still working on vaccines. And then, COVID came, and there's vaccines out and they're already marketed. So what's the difference is there was a world effort. There was governments got involved, lots of funding. There was a lot of funding that was put into all this. Industry got involved, philanthropy got involved. There was many research collaborations. Researchers shared a lot of data. A lot of the publications became open access, so we're able to access the publications, anything to do with COVID, all the publications are open access. So there was this worldwide effort and sharing of data and sharing of information. And most importantly, there was a lot of funding that was put into it. And basically, if there is funding, you could fast track just about anything. So what has COVID learned? What have we learned from COVID is that it's actually paved the way for new immunotherapies to be tested in a fast and sort of quick manner. So, you can, if there's funding available, you can develop these vaccines a lot quicker. No shortcuts will be made, but, they can be made quicker provided there are funding. I mean, a lot of the clinical trials I did, we were involved, we'll do a phase 1 trial, we'll complete that, we'll analyze the data, we will publish it, then we'll apply for grants. Half the times we don't get the grants. When we get a grant, by the time we get the grant, there goes another 1-2 years. Then we will start the next phase. So things just took a long time because it took a long time to get funding. But I think with COVID, we've learned a lot that, when there's funding and there's this global mass effort, we can develop new treatments. So my take-home message isn't, COVID is a pandemic, but in my opinion, I think cancer is a pandemic, and a lot of these other chronic diseases are pandemics too. I mean, why can't they be pandemics, and why can't we all do something similar and get some of these immunotherapies out a lot quicker? So that's what I'm going to finish up with, and I'm from Victoria University in Melbourne. If you are ever in Australia, please come and visit me, and it'll be great to show you around. Thank you.
Thank you very much. Thank you very much, Dr. Apostolopoulos. Our next speaker is going to talk about a topic that is relevant to our research and clinical trials, irrelevant to whatever field we're working on. So it is really my pleasure to welcome Dr. Konstantin Fragakis, a professor at the Department of Biostatistics at Johns Hopkins University. Thank you very much for being here with us, Dr. Fragakis.
Very much. Thank you very much for the invitation, and I'm looking for a pointer. Is there a pointer? Ah. Okay. Oops. Okay. Yeah. So let me see how much my time. Okay. My talk is going to be different from what I've seen, and that is probably because I am dealing with the interface of research and numbers and medicine. But I hope we can learn something, including myself, if there is some interaction about this. So I will talk about 2 of the 3 bullets. The second, which is about how we can tell if one effect is mediated by another, and the third, which is a little bit more involved. So let's see if I'll have time to do that. So I'm going to skip this one. Right. So to lay down some context, suppose that we are dealing with patients who have primary progressive aphasia, and I am the statistician of some of these studies conducted by Kirana Tsapkini over there. And so the the studies are trying in general to see if passing some little current through the brain at particularly chosen areas can help those patients in clinical ways, for example, to improve their language, and also to improve, hopefully, how the connectivity of their brain loci is at that time. Okay, so in a series of works, it has been shown that indeed the treatment that I just said, TDCS for transcranial direct current stimulation, when coupled with some language therapy, behavioral therapy, is improving both. Let's see. Yeah. A and B. Okay. One somewhat more challenging question is whether these two effects are connected. You may say plausibly they would be connected, but you would like to see if there is any evidence for that. So in particular, let me phrase this question here. Suppose we can look at the patients for whom TDCS versus the sham is just the behavioral therapy, has no effect in the comparison, has no effect on brain connectivity. For those people, would like to see if there is still an effect on language performance, because if there is, then we would know that TDCS is affecting or improving language performance in pathways like beyond the brain connectivity, at least that we measure. Okay. Now, I'm going to use this chart here, or flow chart, to say what is the standard trap. You see, I'm talking about traps for trying to answer this question, and then just mention a little bit about how to address that. So here is the two treatment groups, TDCS and sham, and in this second column over here, I show that there are some people among those who are assigned TDCS who develop a decrease in brain connectivity, and some people who develop an increase in brain connectivity. And the same over here. Maybe not the same groups, but no. Okay. So since I'm looking at this effect over here, what a standard method usually does is it takes this group, this whole circle over here, and it compares it to this group. The idea being that when we do that, we compare in both cases people who have an increase in brain connectivity here and here after treatment. And among those people, we are trying to see if TDCS versus sham has any differential outcome on language. So when I say here no effect, I don't want us to confuse it with the fact that I'm gathering these people and these people for whom there is an increase. The increase is in time, but there is no difference between these two groups, okay, in terms of brain connectivity. Okay, that's what a usual analysis does. It compares these two groups, and if you find an effect, you say, yes, there is an effect, If you don't find effect, you say, no, there is no effect. Okay? I'll just be very blunt here that conceptually this is wrong, because if TDCS has any effect on brain connectivity, and we know it does, because I said that at the beginning, then these people here are more than these people here, and moreover, they are not just more, but they are different, probably, plausibly. And so when we compare these to these on their language performance, to see if there is any effect of TDCS versus some, we have broken the balance that is that had been created by the experiment. So you may say, well, what can you do? One thing that we can do is try to find among those people who are the subset who are also here, and then within the subject, like a matching group by group, compare their language outcomes. So I don't have time actually to go over the third part that I said, or the first, but I wanted to take some, I chose to take some more time here to show you how the language that we use, like say whether it's English or whatever, that doesn't matter, can be easily setting traps to us in how we interpret a scientific question to an analysis. And this is one example for that, and those other two situations that are in the slides are two other examples. So thank you very much for the attention.
So, Dr. Fragakis, if you would like to go over the additional slides, please do.
I can, but then I'm going to be asking you the same question. But I will try. Maybe in 5 minutes I would try to give the third one, because it is actually very tantalizing to me. It has been very tantalizing to me, and I hope I will share a little bit of that.
That's great. Thank you.
Yeah, sure. But now, as I said, this third part is even a little bit more different than what you may have seen, unless you have been reading something like Roger Penrose's books. Okay. So there has been a recent focus, or increased focus, on whether the brain in general, when we think cognitively about, or even logically, let's say, whether the brain is a programmable machine. There are some people who, like Lucas and Penrose, who claim that the brain can actually solve problems that no machine can, at least as we understand machines now. And this uses Gödel's work. Gödel was a mathematician in 1932, importantly. Now, if this is correct, so the brain can actually solve problems that no computer can, we would like to understand how the brain does that. Because then we may have at least some idea of generalizing at least such behavior within ourselves and achieve better things. Okay? Now, in order to comment on that, I have to, and I want to go a little bit over what is this Gödel result. Okay? And to do that, I'll try to compress most of the things, but and I apologize in advance if some of these things will not make much sense, but I'll try. I'll see in 3 minutes or something. First, consider some research hypothesis, maybe in medicine or something. Is a surgical procedure in the heart, blah, 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 going to treat arrhythmias for more than X% of the patients with arrhythmias, okay? So by X, I mean, I can put here 50%, I can put 30, I can put 80, whatever, okay? So this is a research and medical hypothesis. Now, there is something interesting that I'd like to invite you to do. Think of all possible hypotheses you can write down: medical, chemical, astrophysical, whatever. You can actually list them 1, 2, 3, 4, 5, 6, blah, 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 not ending, but you can put them side by side with the numbers. And it's an interesting exercise to try to do the first few. I'm just going off and the way you can do that is to see what you are typing. What is this letter that you type in the keyboard? It's the 32nd. What's this? It's the 23rd. So you can actually code all this precise statement as a number that is the number of this guy on the keyboard, the number of this guy, the number of space. So in this way, anything you can think about, even like Shakespeare and whatever, has a number. And from that number, you can actually go back and improve it. So now I'm going to do something interesting here. Okay, so think of all these hypotheses placed as rows. First hypothesis, second hypothesis, third, etc. And they are, each of them is a function of a number, just like the X%. So for example, this hypothesis is the square root of X, a whole number. Let's call that hypothesis so is the square root of 1 a whole number? Yes, true. Is the square root of 2? Is the square root of whatever of 4 a whole number? True. Is the square root of 5 whole number? False. Okay. So for every hypothesis that I said, and there, you can list them. You can envision here a true or false. You may not know all these answers. Okay. And now it's easy for a moment to think about that they are just mathematical hypotheses and not medical. Because the result, and it's a remarkable result, is the same. So here are the hypotheses. For example, this is the hypothesis 2 on number 3, okay, etc. And this is the blue table. Now, what we want is to ask if we can prove a hypothesis using the scientific method. The scientific method is you start with certain principles, you make a question, you go and get data, you do it, and you move on, okay? So it's a built up like a pyramid. So, for example, can we prove that this is true? Yes, I can prove that the square root of 4 is 2, is whole number. So I would put here a yes. Here I would put also a yes, but some of these cells may be no, which would mean I cannot prove it. Okay, I don't know. Maybe there aren't. Maybe there are. Okay. So think of flipping now this, and instead of whether this is whenever this is a true, you put a false. So this table would be the non-provability of the hypothesis in blue. Now I think two minutes, I would say. Okay. So I hope I haven't lost you, because this is the important thing, and it has to do with our language. Take this diagonal here, which says this is a hypothesis, and says the hypothesis 1, here is not provable. This says the hypothesis 2 is not provable. This blah blah blah. So take this line here, which for different positions is a hypothesis like this. So because these are words, I say, you know, I can prove this. This line here, which is about non-provability or probability of these guys, this line must be some horizontal hypothesis. I don't know which one, maybe the 5th, the 7th, but let's say it's the 7th. It has to be one of a horizontal thing, because this lists all hypotheses, if you remember. So then what you get is that for the cell here that is common, and that meets, that's the meeting point of this diagonal and this horizontal line, there is something very special. And what's special about this hypothesis here, this guy, is that if it is true, then it must be not provable, because it is the same cell. This cell here, from the blue stuff, says this cell, is it true or not? The fact that this is the same cell as this also says that the wording, whatever that guy is, or the wording of whatever this hypothesis is, must be the same wording semantically as saying that it is not provable. And so you come to this conclusion that if this question is true, it's not provable. Now, that immediately tells you that it must be true and non-provable. I will not say why, but you can think that if this is false, then this is false, but this being false means that it is provable. But if it is provable, how can it be false? So it must be true. But if it is true, it's not provable. Okay. So now I'm jumping out of the math, and I'm coming to this conclusion that has been observed since 1932, that here is an argument that finds a hypothesis without even telling you what it is semantically, but showing that it is true, but it cannot be provable by the scientific method. Well, how did I prove it? That's the question. I know I did, because you have built in the fact that it cannot be by the scientific method, but you have proved it. So the remarks about this is that it seems the brain can see certain results being true, even if they cannot be proved by the scientific method. Some people say, well, no, and if you Google a little bit and say, but this, yes, I forgot that you have to have all the axioms being true, whatever. Yes, but you always have to do that, okay? So even in the axiomatic method. So the effort then is to try to characterize what is this way. In this way, my effort to explain that is here, is that the language that we have provide, because the language we have is the same as describing something, and whether we can prove it, there is an overcrowding of the facts that can be proven. There is an overcrowding. Overcrowding means this thing about probability must be somewhere here. And this overcrowding creates a relation between what is true and what is provable without actually knowing what they're talking about. This is a way of understanding this, and it is important, I think, to try to increase this understanding. Penrose, even after his Nobel Prize, is working on establishing a connection with this method and quantum mechanics. And there is also a nature of teleology, which I don't have time to go through in this method, which is very different from what we usually do as step-by-step analysis. So the trap here, we say what the trap is. The trap is to think that our brain necessarily is a machine. And of course, the trap is to claim easily also what it is actually doing, if not that. And there is, I think, a lot more to understand about it. Sorry for going over. Yeah, thanks.
Thank you very much, Dr. Fragakis. Thank you for this very interesting, illuminating presentation on a topic that is difficult to grasp. For that, I'd like to, we're concluding our session. Thank you all for your attention, and we'll see you in the next session. Thank you.
So welcome all to our afternoon main session in this year's AMF, which has the title Quo Vadis, Socioeconomic and Ethical Determinants of Modern Medicine. As you'd appreciate, this topic has started from the morning sessions regarding ethos, which we consider as the key topic of this AMF, since the main logo is when Hippocrates meets Aristotle. That means when all the doctors who have given their Hippocratic oath coming from Greece actually come in Greece and they meet the great philosopher Aristotle. After his name, we have the university here, and we are very happy that we introduce to all modern medicine and all technological aspects and issues that are discussed, ethical issues, as we said. So in this session, I have the honor and the privilege to co-moderate with Elias Mosialos, who is Professor of Health Policy in London School of Economics. Unfortunately, he couldn't join us in person, but we always enjoy to co-moderate with Professor Mosialos. So the floor is yours, Professor Mosialos.
Thank you very much. Thank you very much, Mr. President, and thank you very much for the kind introduction of the theme. I wouldn't like to waste much time on speaking about the topics now. I will offer some concluding remarks, and I would like to start our session with our eminent Vice President of the European Commission, Mr. Margaritis Kinas, who is quite well known in the field of medicine, particularly for his massive efforts to make the vaccines available to the European citizens, and also for coordinating the European Commission's effort in the area of health policy during the 2.5 years of the pandemic. We're all grateful to you, Mr. Kinas, and it's a great honor and pleasure to have you here. And without further ado, I will ask you to give your introductory remarks.
Dear friends, dear professors, Agapite Ilia, it's an honor to accompany you this afternoon and to see so many familiar faces today and yet another AMF. You know that you can consider me a friend of the Aristotle Medical Forum. I was with you last year and it's a privilege to be with you again. The forum could not carry a better name and a better title this year when Hippocrates meets Aristotle. Aristotle especially is from this region. I would say a compatriot of mine from Chalkidiki, but also Hippocrates together with Aristotle can be considered as the godfathers of evidence-based medicine. And it is in this Aristotelian spirit that we meet today to discuss the future of health policy. Since we met here in our beloved Thessaloniki last year, many things have happened. And many things have happened, if anything, that further prove the centrality of health and public policy in our society. Last winter, many European cities were still under severe restrictions, business were halted, travel interrupted, and the health sector under extreme pressure. In these unprecedented times, the European Union also delivered unprecedented measures. Probably the most historic, symbolic, and at the same time effective amongst these measures was the successful deployment of the EU vaccine strategy, a measure that saved millions of lives and allowed the society to go back to normality. This has been a great European success story. We designed, implemented, funded, and distributed the biggest vaccination program in the history of mankind. 5 billion doses of safe vaccines, but also 2.5 million doses that were exported from the European Union to the rest of the world of which 500 million doses for free. We were not only the champions of solidarity within the European Union, I dare to say that we were also the world champion of solidarity, since no other bloc in the world could match our international solidarity effort on vaccines. But let us be very clear. We are living a false normality. As we speak, Europe is again hit by a new wave of infections. We are not done with COVID-19. You know this better than me. And all the attention needs to remain in managing the pandemic. And whilst we still struggle with COVID-19, other health threats are emerging in parallel. I'm sure that many of you are involved in addressing the surges of hepatitis and of monkeypox. The two diseases are spreading and their symptoms can be severe, especially for the vulnerable. But we also face another type of virus. Tragically, a new threat has emerged 5 months ago, Russia's unlawful and brutal military aggression in Ukraine. Peace and security, which we were taken for granted on the continent, are among the most fundamental determinants of health and well-being. The people of Ukraine are struggling just to stay alive. Their communities are being destroyed, and many are denied basic access to hospitals for survival, but also to elementary services like food, water, essential medical and public health services. Against this background, the European Union have opened our arms to Ukrainians. We have taken the most powerful sign of solidarity. For the first time ever, we have granted blanket protection to all those fleeing the war with no home left to go, including access to hospitals and healthcare. And in the face of ongoing health threats and challenges, no actor deserves more praise than you, the doctors and the medical community. We were applauding you back in March 2020, and we keep on doing so. Your handwork, dedication, and tenacity is saving lives and protecting people. And this is an inspiration for all of us, both in Europe and worldwide. Dear friends, ladies and gentlemen, the right to health is one of our fundamental values, a symbol of what the European Union stands for, an intrinsic part of what we call a European way of life. To make it a reality, there are several lessons that we must keep in mind if we are to stand up to our commitments and go stronger as a union. First, member states must strengthen their healthcare systems and overall resilience, because we need a strong healthcare sector where practitioners can excel and patients can be effectively treated. This is crucial to cope better with current and future health threats. We also need preparedness and surveillance. We need more alert, and we must monitor closely the behavior of viruses and cross-border health threats, their evolution, their impact, the vaccine effectiveness, and therapeutics. Both the pandemic and now the war are teaching us one core lesson. In Europe, we must secure our supply of critical resources and strive towards European strategic autonomy. It took us a pandemic to realize that we do not manufacture masks, ventilators, to realize that our raw materials for pharmaceutical products are not produced in Europe. We need to correct that. We cannot experience these type of problems ever again. And from energy to health, the lesson for Europe is very clear. We must rely on our independence and our strategic autonomy. To make this a reality, we have been working hard in the last 2 years and a half to bring about a new ecosystem, a new set of structural changes that would make Europe to decide faster and act better in the area of public health. We call these measures a European Health Union, which is under construction, but is already a tangible construction. It's not a slogan, it's not an objective, it's happening as we speak. An obvious structural change is our decision to strengthen and better equip our European agencies in the area of health, the European Medicines Agency and the European Centre for Disease Prevention and Control. We also need to ensure the readiness of our union with medicines, medical supplies that can be used in case of health emergencies. And for this, we have created HERA, the EU Health Emergency Preparedness and Response Authority. That's the European equivalent of the US BARTA. And we're doing this as a proof of our determination to act before threats hit us. And an example is our current work through HERA on monkeypox vaccines. Another aspect is to improve the coordination of our member states across border. We need more effective rules to govern risk assessment, preparedness, early warning response, and the work is now being completed in this front. And finally, we're putting money where our mouth is. We have created a new structural trans-border program for health. We call it EU for Health, which is the most ambitious ever EU program for investments in health with a trans-frontier dimension with an overall budget of 5 billion euros. Beyond the pandemics and the viruses, we're also addressing with renewed force one of the main causes of death in Europe, cancer. With our Europe Beating Cancer Plan, we're coming up with proposals to tackle the entire disease pathway: prevention, detection, cure, and survival. We want to bring hope to patients and families, as cancer is a disease that affects us all, directly or indirectly, and cancer will remain a top priority. Fighting cancer will remain a central European priority. And last but not least, there is no effective health policy in Europe without a new drive for digitalization. In May, we have presented a new groundbreaking proposal for the creation of a European health data space. The objective is that people, researchers, and policymakers have better access to the wealth of health data across the European Union in order to improve both healthcare delivery, but also boost innovation. This is a flagship project, a first in the world, and landmark for all of us, in particular for patients and practitioners. Dear friends, as you can see, there is a lot going on all fronts, and still all this is not yet enough for a strong and resilient health union. Looking ahead, we should continue making progress at least in 3 additional policy areas. First, we need a vibrant, a robust, and fair pharmaceutical sector. This implies 2 things, 2 policy objectives: accessible medicines for all and affordable conditions, but on the other hand, ensuring that the European pharma industry remains innovative and competitive, a world leader, as it is now the case. These are not antagonistic objectives. On the contrary, both are possible, and we should make them a reality. They will guide the important legislative proposals that we are preparing and will present by the end of the year to revise the existing pharmaceutical legislation of the European Union. Secondly, we also need to modernize our health systems. This implies reforms and costs a lot of money. But member states will not be alone in doing this. Europe is once again extending our hand and injecting money through a new instrument, the Recovery and Resilience Facility. This new instrument brings once-in-a-life opportunity to use the massive amounts of EU funding for the creation of more resilient health systems. Finally, our European Health Union can only be strong if our actions are projected and taken up worldwide. For this reason, we will define our health priorities in a new global health strategy to be adopted in the autumn. And we will continue to advocate for a strengthened World Health Organization, fully equipped to play its leading role in dealing with health cases on a global level. And I'm delighted that the WHO representatives will also address the AMF later today. We also want to foster in this quest for international cooperation a new pandemic treaties at global level, because you can imagine that health threats do not stop at our European borders. Dear friends, let me conclude by saying that as the world is changing, the European Union is changing too. Both the war and the ongoing health threats show the urgency of further strengthening our coordination, the robustness of our system, and our resilience. We also need, more urgently than ever, the European project to be based at strong, new, reinforced basis of independence. The era of our innocence in Europe is well over. Let us honor this call from society, in particular younger Europeans who demand a Europe of health, exactly as they expressed it very strongly in the Conference of Future of Europe that has been concluded recently. And let me reiterate my appreciation and congratulations to your work, doctors. You are the linchpins of our health union. We need your frontline and on-the-ground experience, and we need this to leverage, to make things happen. And the organization of this forum, yet another year, proves how much your sector wants to play an active role in designing the future of our health policies. We meet here as Hellenes, as Europeans, but also as citizens of the world. And we share collectively, I believe, the wish for societies that are at the same time fair, equitable, and prosperous. And as Europeans in particular, we reiterate our belief for this continent to live in peace and in security. And health, recovery, resilience, autonomy, and independence are intrinsic, central parts of these aspirations. This is the model of society we stand for. This is the European model of society that we need to guide our lives. Together we can promote it, and I'm sure we will. Thank you again for your welcome, and I wish you all the best for your Congress.
Thank you so much, Mr. Vice President, for this great presentation. I have to admit that I've been a student of European policy developments in the area of health policy for more than 30 years, I would say. And it is now the first time that the Commission has presented such a strategic plan, which is not only a plan, you have already started implementing concrete measures and policies to improve the health of the European population. Many congratulations on outlining the strategy starting from resilience, how we can address better the problems of the Europeans, the health problems of the Europeans, and be prepared. Preparedness was the second strategic point you've made to address the next pandemic, and also complete our efforts to address the existing and ongoing pandemic. You made important points about improving the agencies in Europe, the EMA and the ECDC, and also establishing the new agency in the form of HERA. This is quite innovative, I would say, following the steps of BARDA in the United States of America, and also making data the cornerstone of the European developments. Without data, we cannot improve healthcare. We all know that. We will be in the dark, and we cannot plan the future. And in particular, our country, Greece, has a strategic advantage here. We have a single insurance fund in Greece, and we can use the data we are collecting in the best possible way to develop policies, but also inform policies and clinical developments in other countries. And you made great observations in terms of the assistance the Commission is going to offer to the health systems in the member states through the resilience facility, and the need to have, if I interpreted it well, a new social contract between governments and the pharmaceutical industry. This is what we need now. You made the point that we need less expensive pharmaceuticals, but at the same time to have a thriving pharmaceutical industry, because it is the European pharmaceutical industry and the US-based pharmaceutical industry which are the pioneer industry in the area of biotechnology in our world. And finally, the European Commission is not an island, and Europe is not an island in the world, and therefore it requires a global strategy. And it's the first time that this strategy is being developed, and I look forward to hearing more in the next Aristotle Forum next year about how Europe is going to be the leading force in the world shaping global health policies. Thank you very much again, Mr. Vice President.
Right. We thank Professor Mosialos for his comments, and also the Vice President of European Commission, Mr. Schinas, for the excellent so far introduction of this topic regarding these very crucial issues. And we will go on to this session with Professor Peter Angelos, who is a Linda Kohler Anderson Professor of Surgery and Surgical Ethics and Director of McLean Center for Clinical Medicine Ethics, University of Chicago. Professor Angelos, welcome and thank you for joining us remotely.
Thank you. It's truly an honor for me to be with you remotely. I wish I were there with you in person. That would be so much more enjoyable, although Chicago's nice in the summer, but still, I would like to be with you. I'm going to go ahead and share my screen and ensure that you are able to see. So please do stop me if you're having any difficulty with seeing the slides or with my audio. I have to say I'm truly humbled to be on this program and following such an outstanding presentation as you just heard. I'm going to take the prerogative of my profession, which is as a surgeon, and ask that we take a few minutes to focus on a topic that's much smaller in scope than what we've heard about, which is critically important global health and European EU, the role in global health. I want to look at the surgeon-patient relationship and why I think that that is so central to the future of surgical care. And so I hope you'll indulge my slightly different perspective, which is, again, I'm going to take a much narrower view. We know quite well that medical ethics is an important topic. Especially for this group at this symposium, we know that the concepts of medical ethics go back to antiquity, and it has been for decades that medical ethics has been important to medical education, and this is now widely accepted. When I was in medical school, I certainly had courses in medical ethics. I read books about medical ethics. There were lectures, etc. Surgical ethics, I would suggest to you, is a much more recent term. And I have a medical degree. I am an endocrine surgeon. I do lots of thyroid and parathyroid operations, but my PhD is actually in philosophy. And when 30 some years ago, I said to people, I'm very interested in surgery and ethics, many people said these things don't go together. I had faculty members tell me that surgical ethics is an oxymoron. These things just don't go together. Nevertheless, I would argue that that is a flawed view, and I'll try to give you some reasons why I believe that's the case now and especially in the future. So we know that there are many, many differences between medicine and surgery. We know there are differences between physicians and surgeons, and there's obviously much that goes into how we all choose to go into what we do go into as far as medical specialties. That being said, let me focus, because I only have a few minutes, let me just focus on a very important topic, which is how surgeons view responsibility. And so that, by that I mean how surgeons view the responsibility that we have for the things that we do to and for our patients. Now, from a philosophical point of view, responsibility is a huge topic, and we could spend hours, but nobody would want me to. So rather than doing a long exposition of what is responsibility and how is it viewed in surgery, let me instead turn to a wonderful book by a medical sociologist, Charles Bosk, a 1979 book entitled Forgive and Remember. So this is the second edition, Forgive and Remember. Charles Bosk, as I said, was a medical sociologist who did his graduate studies at the University of Chicago, where I'm now on the faculty. And he decided to study how surgeons are taught and how surgical residency helps surgeons to learn how to deal with errors and, as is in the title, managing medical failure. So Bosk spent lots of time at surgical morbidity and mortality conferences. And he wrote, I think, a beautiful statement of a very succinct difference between how physicians view responsibility as compared to how surgeons view responsibility. And I will just read this to you because I think it's really helpful as a backdrop for my future comments. When the patient of an internist dies, the natural question his colleagues ask is what happened. When the patient of a surgeon dies, his colleagues ask, what did you do? By the nature of his craft and his beliefs about it, the surgeon is more accountable than other physicians, and he also has much more to account for. Now, I won't spend a lot of time focusing on this distinction, but I believe it is very accurate. And I do believe that this notion, this concept of responsibility, significantly shapes the way surgeons think about our relationships with patients and our activities in surgery. Now, why examine the surgeon-patient relationship? I mean, what's different about the surgeon-patient relationship and other relationships between physicians and patients? Well, a number of things I would suggest are important and worthwhile to consider as distinctive. First of all, when it comes to surgery, harm is absolutely necessary in order for there to be even a possibility of healing. So we cannot, as surgeons, impact the health of patients without doing things that are potentially harmful. So every operation is in some sense a potentially violent act. And patients have to be willing to go through that in order to have the possibility of healing. Additionally, surgical interventions are illegal in any other circumstance. So again, I mentioned I am an endocrine surgeon. So I do many thyroid and parathyroid operations. And so as my children, when they were younger, told their friends, they said, our dad cuts people's throats for a living. Now, that sounds bad, but that's in fact an accurate statement. And what I do, making an incision in someone's neck, I would go to jail for, were it not for the fact that the patient has given me informed consent and I'm in the operating room. So in that sense, I think surgical interventions are quite different from many other interventions in medical care. Additionally, it's an intensely intimate physical relationship. As surgeons, we know things about our patients that they will never know. Patients frequently ask me and other surgeons to take pictures of the tumor because that's what they really want to see. They never get a chance to know what that looks like. In addition, it's frequently a very short time frame in which we have to engender the trust of our patients. I may meet a patient 30 minutes, 45 minutes, an hour later. I'm asking if they're willing to allow me to do something that's potentially very harmful for them if there's a complication. And then finally, I would say consent for surgery, which is, again, the basis for the ethical practice of surgery is informed consent. Consent for surgery is not a checklist, but a manifestation of the tremendous trust that our patients place in us. So let me turn just for a moment to informed consent for surgery, which again, this is a very widely discussed topic in surgery, and it's very clear, it's not news to anyone that the patient has to have the capacity to make decisions, or we involve a surrogate. So we do not ask comatose patients to decide for surgery. We go to a family member, we go to a surrogate. In addition, we traditionally, as surgeons, explain the risks, the benefits, and the alternatives of an operation. And then we ask the patient for permission. Now, this is widely accepted. But what's very interesting, and I believe has not received quite the attention that it should, is how should the quality of the informed consent process actually be determined? Now, traditionally, the assessments have been, does the patient actually know the risks, the benefits, and the alternatives that the surgeon explained? And lots of studies have looked at this. Multiple studies, though, show that most patients do not remember what their surgeons tell them, even a few minutes after the surgeon explained things. Nevertheless, there is, in many studies, a high degree of satisfaction in the informed consent process by most surgical patients. So this necessarily leads us to ask this question: how can there be this discrepancy? Patients remember little of what we tell them about risks, benefits, and alternatives, yet they remain highly satisfied with this interaction, the informed consent process. So from my perspective, we have to ask the question, is information transfer the true measure of success of informed consent? In other words, is it that I am transmitting information to my patient? Is that really the most essential ethical aspect of the informed consent process? Now, not to spend a lot of time, since I have written about this and encourage those who may have an interest to look at some of those publications, I would argue that patient trust transcends problems with memory. And that even if someone doesn't remember what the surgeon told them, if the patient trusts the surgeon, that, to me, is an effective informed consent. And therefore, I would argue that it's actually patient trust that is the basis for informed consent, rather than that I have conveyed information in a robust way to my patient. Information is good, it's valuable, but it's important that patients trust us. And that, I think, is the most essential ethical aspect. And in fact, data shows that surgeons trusted by their patients are less likely to be sued by their patients despite having complications. Now, let me then summarize some of my thoughts about informed consent. I do think that informed consent should mean much more than this checklist of risks, benefits, and alternatives that we present to patients. Certainly, good data is essential for informed consent, but it's not enough. It's not enough for a surgeon to be able to tell a patient the risks of a complication are 1% or 10% or 20%. That's important, it's essential, but it's not enough. Patients have to be willing to allow their surgeons to place them at truly significant physical risk for the possibility of benefit. So I would suggest to you that technical surgical ability is absolutely necessary for surgical excellence. It has been in the future, it will continue to be, it has been in the past and will continue to in the future be necessary for surgical excellence, but it's not sufficient for the future. And so let me just turn in the last few minutes to some challenges as I see them for the future of surgical care. So first of all, surgeons are primarily paid to operate on patients and not talk to patients. And so I think this is a fascinating financial incentive model. So in the emphasis on surgical volumes, how many operations we do, how efficiently we convert patients that we see in the office to patients who are having surgery. That efficiency, the drive for efficiency, the drive for increasing surgical volumes drives many, many surgical departments, many medical hospitals, medical centers, etc. The secret truth, though, about surgical success is that one cannot garner the trust of a patient without having significant high-quality communication skills. And thus, I would argue that the challenge for the future of surgery and future surgeons is to be a complete surgeon. So what do I mean by that, to be a complete surgeon? Well, as I said, technical expertise is necessary, but it's not sufficient. A complete surgeon is a technically excellent surgeon who's also a great doctor in the broader sense of the word doctor or physician. Surgeons, I argue, must withstand the temptation to become purely technicians and thereby cease to be true physicians. And this is a risk because medical centers want to increase the volumes of operations. And so pushing surgeons to focus their attention in the OR, not spend a lot of time talking with patients preoperatively, postoperatively, that I think is problematic for the future of surgery. There are a few added challenges for today's surgeons, and I think we have to acknowledge these. The central question for surgical care has changed. The central question used to be, what can we do for this patient? And my father was a surgeon. For his entire career, this is what he focused on. What can we do for this patient? Today's question is different, though. It's what should we do for this patient? And that's a very different question. So there are many things that we can do. The possibilities for surgical care, critical care are astronomical. But what should we do for this patient? That is a question of surgical ethics. And that is something that technical ability alone will not help us to answer. In order to answer the question, what should we do for this patient, we have to communicate well with patients, we have to understand their values, we have to ensure that we're not bringing our biases as physicians and surgeons into that relationship. And that will be critical for the future. So I'll leave you with very few conclusions. I think surgeons have in the past and will continue in the future to need to consider the ethical dimension of patient care. So what do I mean by the ethical dimension of patient care? That's communication, that's the relationship with our patients, that is the ability to engender trust. By deliberately focusing on this ethical dimension of surgical care, I believe surgeons will be better physicians and show others that they are more than just technicians. And again, I think this is central to the future of surgery. Finally, I would say surgeons must be experts in the ethical care of their patients. We cannot outsource this to others. It must be surgeons that take a leadership role in this area. And thus, I would argue that the surgeon-patient relationship is the key to the ethical care of the surgical patient. So with that, I want to thank you for the opportunity to present virtually. I realize that my perspective is a much narrower focus on the doctor-patient relationship, but I really appreciate having the opportunity to participate in this conversation. Thank you.
Thank you very much, Professor Angelos. From what you have said, it seems that what we advocate is being confirmed by you, that the concepts of ethics go back to antiquity, and we believe that as offsprings of Hippocrates and also a lot of philosophers in Greece, we do have, as yourself, coming from a Greek origin, our inheritance that we can translate it into clinical practice. And we are very glad, I'm a surgeon as well, I'm a heart surgeon, that you balance between surgery, clinical practice, and philosophy and ethics. That's excellent. So what you have said is surgical ethics is something relatively new, and the major, let's say, message that I would take home from your lecture is, from what you have said, nowadays is not the question what I can do for my patient, but what I should do for my patient. This is excellent. So on this background, I have just two questions regarding your talk, and mainly the informed consent that you have focused. And we as surgeons always mind about giving data. So regarding to this, let's say, this thing, what do you think, what is the data that I need to inform my patient? Is it the international data, I mean, evidence-based medicine data? Is it institutional or national data, or it is my personal data? How can I overcome the bias by giving data to the patient? This is the first question.
Yes, thank you very much. That's an excellent question. And I think that it strikes me that it is important for us to share with patients what are sort of the national rates of complications. And depending on, maybe it's, if you believe that there are significant differences within one's country, then perhaps regional rates are important. But I think this idea of what ought a patient in general expect are the risks, I think that's something that we absolutely need to share with patients. But I do think that patients also deserve to know what our individual rates are. And so if my rates of complication are actually higher than the national rates, well, that's critical that a patient know that, because then they need to weigh that into the decision making. Maybe it's easier for them to see me, a local person, rather than travel a long distance to see someone else with lower rates. But if they don't have this information, they cannot be empowered to make that decision. So I think that that's really critical. Now, there is a tendency among surgeons to want to share data when it's better than national averages and not share it when it's worse. And I think that this is where we have to depend on our own self-reflection and personal integrity. We have to share both good and bad with our patients so that, again, we're empowering patients to make good decisions.
Right. So your answer brings me to the next question. I was sure that you'll reply like this. So if this is the answer that I should be honest with my patient and give both if I have a discrepancy between the average and my practice. So is this, when we say concern form, is this an ethical approach or it's a medical legal approach that we are doing here?
Yes. So I think that, again, an excellent question. There isn't clear overlap between what's ethical and what's legal. No question that those things vary. And, you know, what's legal is going to depend on, at least in the US, different states have different laws. There isn't you know, there are some national laws, but there's lots of individual state and local laws. I would say that, to me, being upfront about what one's personal experiences and what the complication rates are, to me, that's an ethical imperative. I think that now in some states in the US, it's also a legal imperative, but not in all. And so, for example, there was a recent court case in Wisconsin where a surgeon was actually found liable because the surgeon did not tell the patient that there are other surgeons in the area that have better outcomes than that particular surgeon. So I would argue that it's an ethical responsibility that's national and international. The legal parts, I think, are a little bit harder because local rules apply.
Right. Thank you. Professor Moscheles, you are now taking the NHS experience for Professor Angelos.
Thank you. Thank you so much for this outstanding and great presentation, Peter. I would like to make a point about the individual versus the global or regional rates, that if we take an international perspective, there is always a risk of what kind of risk adjusters we're using, because it's not just the crude rate. We do need to have patient risk adjusters. And I would say also, we do need to have also supply side risk adjusters, because if you're in a small surgical unit just supported by a couple of nurses, and another person is operating in a big university setting with 30 surgeons and 100 nurses, then inevitably your rate is going to be much worse if you're in the rural area, for example. So therefore, some acceptable, if I may call them, inequalities in terms of reporting rates should be justified in this particular case. But I would like to ask you another question going beyond the point. You emphasized that consent is much more than a checklist, and we do need to have a more holistic perspective in terms of developing the new surgical workforce. But what about the system perspective? There is one issue in terms of trust between me and you as my surgeon, but I'm a well-educated academic. I can look at the literature. I guess many American well-educated people will do the same. What about my uncle who is living in a rural area, does not have access to the internet, so there are all these inequalities in terms of digital inequalities, has no information. What can we do in this particular case? And also, what can the system do to improve the trust? So we're transferring quite a lot of responsibility to the individual doctor here, but are there any system perspectives we can consider? And then based on that, talking about the holistic surgeon and taking the more comprehensive perspective, surgery has evolved tremendously over the past 30 years. In terms of the techniques, they're less invasive, they're more lenient, more efficient, but you still have many of your colleagues who are not following up innovation. Old habits die hard, and we know that. So what is the ethical perspective regarding the surgeon who is not failing, I would say, to adopt innovation? And a final point, which is global. Now, in many countries, as you know better than me, there are no surgeons, or there are only a few surgeons. So what is the ethical dilemma there? You pose the ethical dilemma. What should we do? Is frugal innovation acceptable there? Is training a nurse to perform surgical operations acceptable in Burundi, for example, or in Malawi? What is the position of the surgical profession in low-resourced areas in the planet? Thank you very much.
Thank you so much. All, truly challenging questions. And I don't know that I've got I don't have an answer for all of them, but I at least give you my impressions. So the first question had to do with this sort of the systemic, the system responsibility versus the individual responsibility. And so I do think that healthcare systems, that there is a clear responsibility to try to reduce the disparities as much as possible by increasing the level of accessibility. So I think increasing the accessibility of patients to surgeons, other physicians who have the expertise to deal with these issues, I think that that's critical. There are many patients who don't have the same educational background that we as physicians have. Nevertheless, I think we have a real responsibility to communicate with people in a way that's meaningful to them. So, if I were talking to my grandfather who, came from Greece with a 8th grade education, he was a very smart man, but he didn't have a lot of degrees. And yet I would need to communicate with him in a way that's meaningful so he can make a choice. So I do think that we should provide as many appropriate educational resources as we can. I think that, when I see patients in person, I can draw on anatomy charts. I can write down what the procedures are. I can give people things to bring home and read, to discuss with their, family and friends. So I think all of these things are valuable. Your second question had to do with what I would call the double-edged sword of innovation. So innovation is great if it's new and it's good, right? And so there's a new thing, and people do it, and if it's good for patients, that's great. Sometimes though, we have what we think are good ideas that turn out to be bad ideas. So the problem with early adoption of innovation is that we may put patients at risk. And so there again, what do? Well, we have to disclose to patients the uncertainty of these innovative things. So we have to be sure they know. But there does come a time when we pass from saying this is a novel technique that a few people do to this is now the standard of care. And so what I think is fascinating is that we never really know when we cross that line. So we never really know when we're going from something that's innovative that, maybe a few people do it. And so if I don't do it, I don't need to talk about it with my patients, to at some point we cross a line. And then suddenly, if I don't discuss the fact that there are other alternatives, that there's this innovative technique, even if I don't do it, then I am not ethically presenting the options. And so that is a real problem for surgery, because I think medicine as a whole tends to be very conservative in terms of change, and surgeons among the worst, I would argue, because we do what we were trained to do. So that's a real challenge. And I think the best that we can do is to, again, say it's an ethical imperative on the part of all of us to stay abreast of what is best for patients. So how do we do that? Well, we have to engage with other physicians. We have to read the literature. We have to go to conferences. We have to participate. And when appropriate, if one's in an academic institution, we have to generate new knowledge. We have to do research, write about it. Your last question had to do with sort of areas where there aren't surgeons, where there's a lack of, where there's a real absolute scarcity of resources, be that resources as physicians, surgeons, and resources, in terms of technology. I think that there's a lot that can be done without high technology if we empower people to do what's possible locally. So again, I think that there are a lot of ways to think about this. So, in the US, there are many surgeons who will go to third world countries and do a week of, sort of a mission work where they do a bunch of operations. And that's good. I would never say that's a bad thing. but it may be even more valuable to train local practitioners to do what's possible in that area so the good work can continue even when the expert flies back home. And so that's where I think we need to be very creative in thinking about how can we do good more than just me doing an operation, but how can I help educate, how can I help a local group provide a care that's better across the board, even when I'm not there.
Thank you. There is another last question from the audience here before we let you go, Professor Angelos. Yes, please.
Hi, Lila Amirali. I'm a child psychiatrist professor at the University of Montreal. And I enjoyed very much your presentation, especially the what should I do for the patient. And I would like to just make a comment about where we are in psychiatry today, where we're looking, what can we do together? What should we do together with the patient? And in fact, one of the alternatives is to have what we call patient partners, patient partenaires, where we partner, with groups of patients, we discuss, and we have them so much in research involved. So every research project that we have to apply to the Canadian Institute for Health Research has to have a group of patient partenaires who are partners with us, who discuss if our outcomes are good enough for them, if they're interesting or not, who can help us translate some of what is our intellectual curiosity, and if in fact producing innovative techniques. They exist in psychiatry as well. So how do we do that with the patient at the center? So regardless of their level of, education often, which is a barrier. So thank you very much. I think it is very interesting how there could be cross-fertilization, because it's a huge question. What does it mean, informed consent? And how far do we go with that? And what do they understand? And how can patients help other patients understand better who have had already the experience? What does it mean in their language? And maybe produce collaborations with and partnerships with our patients in the way to their healing.
Yes, thank you so much. That's really a fantastic comment and suggestion. And, I have no experience with this concept of having a group of patients participating in sort of thinking about what are the appropriate outcome measures. That sort of engagement, I think it would be great, and something that I have really not seen done, at least in the US, when it comes to surgical research. And it does strike me that it's sort of fascinating that there's this interaction, because I do think that there's a strange sort of synergy between psychiatry and surgery, in that both are quite invasive. I mean, quite invasive in different ways. But nevertheless, I think, it sounds like we have much that we could learn from you. So I agree. I think this idea of cross-disciplinary learning and cross-pollination is fantastic. So thank you for the comment.
Right. Thank you, Professor Angelos, for being with us. And I give now the floor to Professor Moschelos for introducing the next two speakers who will join the same lecture. Thank you, Professor Angelos.
Thank you.
You have to unmute, Elias. You have to unmute, please. We don't.
So
hear.
I'm going according to the program. Our next speaker is Professor Aristidis Moustakas, who is a professor of medical biochemistry and tumor biology at the University of Uppsala in Sweden. Thank you very much for joining us today.
Yes, can you hear me?
Yes, we can. Yeah.
Thank you so much for the kind introduction. Will it be okay if we also see Professor Kafazadzopoulos, because we have a common presentation?
Yes,
we will talk
is
with
joint.
each other.
Yes, definitely. Yes, Professor Iordanis Kafazadzopoulos, who is a professor of human-computer interaction at the Department of Information Technology, the same University of Uppsala in Sweden. Thank you, Iordanis, for joining us today.
Thank you. Great, thanks so much. Now we have to share our presentation. Bear with us just for a second. I hope it comes in front of you. Is it all right?
Yes.
Great, thank you. So I would like first to start by thanking, in particular, Kyriakos Anastasiadis and George Tsoulfas for actually placing us today in this very fantastic session. It's a true honor for me personally, and congratulations for AMF number 2. I have been watching it since this morning, and it's great. Now, together with my good friend Iordanis, we chose to discuss precision medicine. And we think that this is relevant to the topic of this particular session. And we became extremely happy and glad because those of you that attended this morning, the Hippocrates meets Aristotle and the discussion about ethos from the very distinguished professors of philosophy, in particular, Dr. Skaltsas and Sfendoni Menzu, you probably already have been introduced to the topic. I believe what we will do in a few minutes here is to take a little bit of a different perspective, and I will play the strong advocate of the new technology. I wish I could present myself in front of you as a robot, and Iordanis will be the more critical, deep thinker. Now, we start with this slide, precision medicine. And why do we show you this slide? Because this is reality. It's not something about the future. This is a photocopy of the front website of our Uppsala University Hospital, one of the largest in Sweden, which also has developed a precision medicine center. And we just read a few of the statements of this website, which has as a purpose to inform actually the citizen of our country. And it says, precision medicine means choosing personalized treatments based on the patient's individual needs. This is a fantastic statement. It actually has inside it what we heard this morning about ethos, about values, and things like that. And then it continues with some technical terms. It says that, we need better diagnostic methods. Better, that means actually that the precision medicine application to today's practice is not yet there. It is developing very rapidly, but it is developing. For example, genome sequencing, I will come back to that. And at the end of the slide, you can read the bullet points. It helps us develop an equal and sustainable healthcare system. I'm not an expert on that. Maybe Professor Moschielos can tell us more about this. Enables personalized diagnostics and treatments, indeed, and this is where my research is trying to contribute to this particular field. Improves treatment efficiency and reduces side effects. This is the hope. Improves the quality of life for patients. Okay, remember that, the quality of life for patients, because the patient who has been treated over the millennia with traditional actually medical practice oftentimes has been abused out of the desperation to save her or his life. So not because the doctor was bad, just because there's no better means. And we already heard some discussions about African countries and their possibilities to develop modern technologies. And final point, leads to positive economic outcomes in healthcare. Okay, by means of advertisement, and I know there is actually a growing community of precision medicine in Greece. This is what we have developed in Sweden. Actually, I'm not part of that, so I would proudly like to present Professor Brandell and Professor Kallionemi. And they have developed the Genomic Medicine Sweden, which is an organization that, as you see on the map of the country, sort of co-organizes 7 regional authorities with the 7 big university hospitals with the respective medical faculties of these 7 universities. And on the other hand, we have this Science for Life laboratory, which together with a very recent, super rich, in fact, donation of a program called Data-Driven Life Sciences, is trying to actually develop what we call precision medicine in Sweden. This is at the national level, and internationally, this particular sort of cooperative organization is making a step towards Danish-Swedish collaboration. And we hope that because the European Union, as we heard in the first talk of this session, is really promoting this type of high quality life. This is really what we have to strive for and not make wars. And so this will expand actually throughout the continent. Now, this is my last slide, and I wrote down 5 points I would like to discuss. And as I said, in many of these points, I am dressed as the robotic type of instrument, like your mobile phone that you hold on your left or right hand, which is helping you actually improving the state of your life today. Point number 1. Personalized medicine applies artificial intelligence to everyday clinical practice. You will hear about this from Iordanis. PM aims at being data-driven and less hypothesis or authority-driven. So again, I'm the scientist. I develop my biomarkers and different kind of small molecular details, which are very necessary for the computerized robot to train itself so that it can give back to the medical society of the Aristotelian University or any other society better knowledge to do its job. Now, this is very interesting because suddenly my traditional hypothesis-driven scientific approach will be actually paired with what is called data-driven. So the computer will actually tell me that maybe some of my ideas are wrong and I have to change. Even worse, for you physicians, you make decisions about the best possible treatment of a disease when you meet at the famous congresses of the International Society of a particular field. We can take as an example a very rare disease or something very generic like diabetes or cancer. And there, the top scientists actually, in association with everybody else, decide about the policy. And now these people also, they have to become a little bit humbler and accept the machine, which will tell them that, you know what, the truth and the fact-based truth of the future may not be exactly what you think about. And this is, I think, a very important ethical point, which is from the positive point of view. It's not that artificial intelligence will destroy us as humans. It will actually make us better humans. Instead of pressing buttons of nuclear weapons or other kinds of weapons in the north part of our continent, we will hopefully get the correct advice to not do that. Point number 2, PM beyond genomes. So there are many people that actually hear about this precision medicine. Remember also our website, it gives us an example, genomic research. This is where the technology is actually booming right now, and it delivers the best possible sort of molecular detail to the physician. But I'm going to give you 2 other examples that actually our precision medicine center is very good at, and I chose this totally different. So most of us, even healthy people, have actually taken a blood test and measure the levels of our sugar. If these kinds of data are actually deposited in so-called registries, and in Sweden we have plenty of those, we can ask now, again, the robot to examine this data and possibly give us a new clue that instead of thinking only of the problem in the heart, we should possibly also consider these weird types of bumps that my skin is developing at the age of 59, which nobody thought that it has any relationship to heart disease previously, but now the computer will help actually the physician of today to give a better type of prognosis, if you like, for future. Listen to this second example, is fantastic, I think. Robotic analysis of a physician's medical notes. Over the past 15 years, every physician in Sweden has to actually painstakingly spend extra time every day in order to copy their private notes of discussion with the patient into the registry. The registry is accessible by the patient and the physician, and under circumstances, other people can access this data. Imagine now, and actually this is being done, there are special applications, apps that have been developed by physicians that now take a look at the words that the physician wrote for a specific patient over the years and comes up with new ideas that the physician, because of lack of memory or lack of time, would never go back and read all the notes of the last 7 or 8 years. Fantastic development. a little bit of critique. Artificial intelligence issues. How will the medical doctor maintain the skill to take the best possible decision, as the website of our hospital promises, on behalf of the patient's health? Again, here we do a great job, I think. First of all, we have started collaborating with the informaticians. We have Google. Google actually is trying to develop medical programs. We have what is called bioinformatic programs. And so we're very, very much optimistic that the future generation of physicians will also be actually very well versed into information technology language. They can criticize or at least think about the function of algorithms. And so life in the future definitely will be better. Financial issues. Precision medicine focuses on small groups of patients. So think about this. The COVID-19 gave us a fantastic example. I mean, so fast, and I humbly bow actually to this fantastic 1.5-year development of the global medical and biomedical community that gave me the opportunity to actually get vaccinated so fast. And actually, this is the reason why I'm not in my home city, Thessaloniki, today, because thanks to this freedom that medicine gave to us over the past 1.5 years, and thanks to the vaccination, I'm able to travel in 2 different places on the same day. And I apologize for not being near you in the room today. Back to my point. So COVID-19 vaccine for the billions of the globe. Precision medicine, new kinds of drugs for who? Just for the individual? That's impossible. But for small groups of people, this causes financial issues. But we know that the pharma, actually the big pharma, has accepted this model and is building on it. So we're very optimistic. In fact, you know, the pharma that is criticized often for only having an eye towards a big pocket of money is actually developing future programs, which in order to lead to the drug actually have to spend a lot of money. They're very expensive, and eventually will help the very few hundreds or thousands of people that have been now clustered in what is called an individualized type of group. Final point, integrity issues. Will PM endanger human integrity, equality? The philosophers of the morning were the proper people to address this. Again, I want to put on my robotic type of dress and bring up 2 things. The things we usually discuss around this is that the employer will discriminate because they have some information from the genome and so on, or the insurance companies will not cover us anymore, you know, because they learned that actually in 30 years, I, for example, well, I will not be alive in 30 years, but possibly in 10 years, I will develop you know, this terrible disease, and then why would they insure me? Well, I'd like to say the following regarding integrity. I'm very happy, and actually my personal data exists in a registry of a huge experiment that Sweden has done. So far, about 80,000 individuals have been scanned, their whole body. I really hope that the molecular identity of some of my blood molecules, which I know very well, or some pictures of my liver and my heart will be actually available to the whole community globally, so that some smart individual, future students of this English-based program of the APC in Thessaloniki will actually develop better treatment. I think I spoke enough. Iordani, the floor is yours.
Thank you very much. So artificial intelligence is very important here, and regarding precision medicine. Well, we may say that artificial intelligence is important for everything, and it's coming more and more in everything, in every sector in society, I think. Well, what we think here is important is a certain problem that we have to consider regarding artificial intelligence and its use. As already Aristides has mentioned here before, is that by using artificial intelligence, which of course is unavoidable, you may have some effect on the thinking of the physician, of the practitioner here, that the suggestions by artificial intelligence are necessary and also very good, and they will be much better in near future, and better all the time. So it will be, I mean, very difficult for the physician to say no to the suggestions coming from artificial intelligence, right? And this opens up something, an area, a problematic area here, that is actually not only about the skill of the physician, but it is much broader. Artificial intelligence is excellent, of course, in as a tool for diagnosis and for therapy. It already is doing a lot of excellent things, fantastic things, but it will do much more in the future. But all its importance lies in the suggestions it comes with, the answers it gives to our questions about what is the disease, what is the proper therapy for a certain patient. And this is what we expect for that, of course. But on the other hand, we may ask what is happening with the thinking process, the scientific thinking process of the doctor. I mean, it is there during whole history of humanity in order to give the answer to what is the cause or of what is the disease, what is this person suffering of, and what we can do, what is best to do. And that was the product of the scientific thinking of the doctor. But no more, right? What happens now is that the algorithm is coming here and replaces this thinking of the doctor. Well, is this a problem? This is a very interesting question. And of course it is. And it is if we think more broadly in the whole society, that what we expect, that we want all the time is to have answers to our problems, and good answers. And artificial intelligence will do that. And when it is doing that, then the thinking of the doctors in this case, and humans in other cases, is not necessary anymore. It replaces this thinking. So it is important for us, especially when we discuss ethical issues, to try to understand how we can handle this problem, because we have a lot of discussion here, ethics and care, ethics and technology. And as we heard this morning, there are many ideas about how we can make artificial intelligence ethical. There are ideas about putting values into these processes or doing other things and so on. But this is something that is not possible, I think, to achieve through such measures or by doing such things, because the basis of ethics is the dialogue, is discussion, is the process of thinking. These phronesis, now we are in this Aristotelian setting here, and that it is the process of thinking, and by that is meaning it is how we can run this process in the right way. And this is phronesis. And this is something, it's not the end result of this process, but we are interested mainly in the result, in the answer coming out of such a process. So the ethics cannot be outside this process. It is the process always that produces answers, the ethical answers. We cannot have them just by accepting the suggestion by the technology. It is impossible to achieve that. through this way. So the question here is then how to design this artificial intelligence and how to use it. And of course, the problem comes here that given that what we want is to have the right diagnosis and the right therapy, in this case of precision medicine or medicine generally. And suddenly we say, stop, we don't do that. Instead, we design and use artificial intelligence in a way as to stimulate the scientific thinking of the doctor, which of course is a solution to do. So we can ask ourselves, is this possible to happen? Should we abandon or don't care about very fast and very reliable and the best possible answer coming from artificial intelligence and instead say, just wait, we will support the thinking of the physician to come up with an answer to this problem? It's not easy. But if we don't do that, if we continue with this, artificial intelligence will be better and better and will give us better and better answers to all these problems, and it will fix all our problems, and our thinking will be obsolete. What should we do then? Thank you. Next slide.
I think we have finished, so I try to stop sharing.
Well, thank you very much, both. Unfortunately, you had a full lecture, so we're running very, very late, and we have an opening ceremony coming soon. So I don't believe we will have time to comment to those wonderful things that you have presented to us. Hopefully next year, Aristide here and maybe Professor Kavadazopoulos, we have you here with us, and then we'll have a panel discussion at that time. But so far, thanks a lot for sharing with us this wonderful lecture.
Thank you.
So, thank you. We'll go on with a pre-recorded video lecture on behalf of pharmaceutical industry from Theodoros Trifon, who is the president of Pan-Hellenic Union of Pharmaceutical Industries.
I would like to thank the organizers of Aristotle Medical Forum 2022, and especially Professor and President Anastasiadis, for inviting me here. Unfortunately, I cannot join you in person. I hope I'll do it next time. The world nowadays is facing a revolution in healthcare, which has been accelerated by the persistent pandemic, the recent technological developments, among others, digitalization, artificial intelligence, iCloud computing, real-world data, the world demographic transformation, whereby we have a combination of reduction of fertility, increase in life expectancy, reduction of mortality, and aging population, the increase in demand for healthcare as a result of increasing urbanization, the creation of middle class in many countries due to globalization, and the climate change. Even though the pandemic plunged the global economy into recession, all estimates regarding the day after convergence on the following conclusions lead to the fact that to the belief that we'll have a strong economic recovery after the pandemic. Healthcare and biopharma R&D will be the big beneficiaries and key drivers of the economic growth. Definitely the short-term environment, of course, is altered due to the crisis resulting from the war, energy and inflation. However, we believe that after this crisis is met, to a smaller or lesser degree, again, the economic recovery will be fast. Now, the main pillars of this development will come from the investment in research and development, the focus on innovation, the emphasis on startup entrepreneurship with the creation of new businesses related to the development of new tools, for example, artificial intelligence, and definitely human resource optimization and training of human resources in new skills. It is clear to us that Greece has the capacity to develop all the above 4 pillars. Of course, it is something that is not very easy, and we need time for Greece to make the next big step forwards, especially after a 10 years crisis. A challenge is the relatively low investment in R&D as a percentage of GDP in Greece. However, during the last 3 years and for the coming years, investment in R&D is steadily increasing, and it will be further strengthened. Moreover, we see much larger foreign direct investment now than in the past, and all this hope will create an ecosystem of innovation and R&D. On top of that, we have some discussions now about the European pharmaceutical policy after the pandemic. During the pandemic, health systems across Europe came across enormous challenges in terms of patient access to treatments, adequacy and smooth supply of the pharma market, and of course of all the necessary treatments for this pandemic. These challenges, which will of course be further increased with the recent developments with the war in Ukraine, will mean that that Europe has to see in a different way and create some incentives for patient coverage and access and market equity problems due to the dependence on imports of medicines and medical consumables from third countries, reshoring of pharmaceutical production on European ground, strengthening biopharmaceutical research with the aim of addressing public health challenges, and mainly strengthening the competitiveness of the European pharma industry within the global market. The discussion that has started will result in December in a new European pharmaceutical strategy, a framework that aims to shield EU countries' health systems, ensure patient access, and safeguard the adequacy of medicines in a new potential health crisis. At the same time, it will encourage investments and strengthen R&D and production in Europe, reshoring from abroad many of these activities. For the next 5 years also, the environment for Greece seems positive because of the very large number of funds that will be available. We have the RRF program, implementation of Research and Recovery Fund, whereby some investments of about 250 million only for the pharma industry have been included into that. We have also the new development law, the program for strategic investments, policies regarding favorable tax treatments of R&D expenses, and incentives of attracting scientific human resources from abroad to Greece. All of these form a positive environment for the day after in health and pharmaceuticals in Greece. Now, I would like to come to some research trends and directions in the coming year. The new challenges created by demographic and epidemiological developments, as well as climate change, require the presence of a strong biopharma industry that at the same time is a significant driving force of the new economy. In the current environment, we meet, first of all, the accelerated approval of new drugs, devices and biological. The number of FDA-approved drugs more than doubled in 2020 compared to 2010. While the development and market delivery of COVID vaccines took less than a year compared to the flu and Ebola vaccines that took decades. Second, the fast-track application of technologies that make the drug production much more cost-effective. For example, the cost of producing biologic antibodies between 1995 and 2020 has fallen by twofold, while yield has increased significantly. Decisive driving factors for the acceleration of drug development and approval are, among other, the new techniques for synthetic DNA production, the development of bioinformatics, artificial intelligence, and machine learning, and all these are supported by collaborative efforts between the industry, private initiatives, and the academia. In example, the Aristotle University of Thessaloniki and the Special Unit for Biomedical Research and Education are forming collaboration with the private sector for tissue biobanking, sample analysis, and production of big data that can potentially feed specialized big data analysis software. Drug development will now largely be guided by in silico analysis, and this will result in both acceleration of the time and the reduction of the cost, going from bench to bedside, ever improving the quality of patient healthcare. Now, what does all this mean for Europe and particularly for Greece? As I mentioned before, there are many significant incentives in place. Second of all, we have, I believe, skilled personnel. We have more than before collaboration between industry, academia, university, and research institution. And also, significantly, we have a lot of interest from foreign companies to invest. We've seen that with the Pfizer unit in Thessaloniki. We've seen that with Microsoft unit in the Attica area, which is by coincidence very close to the new Life Science and Innovation Park of Elpen. And also many other companies now are interested to invest from abroad in Greece for big data, for clinical trials, and for R&D investments. We have seen that the stronger the pharma industry and the healthcare sector is in Europe or in Greece, the more it safeguards and shields the community and the patients in times of crisis. So this is very, very important. And of course, the future of us being able to be more self-sufficient lies, as I mentioned before, in several factors. Basically, is the rapid increase in R&D spending and the increase in competitive funding from EU program, the strengthening of the startup ecosystem, the industry-university interaction and collaboration, and of course, the whole ecosystem around innovation. Based on all the above, I am confident and positive that in the next years, we'll see significant investments that will safeguard the Greek population, the patients, and also increase the innovation footprint in our country. Thank you.
Okay, I think last but not least is Dr. João Breda, who is a special advisor to the regional director of the World Health Organization office in Copenhagen, Dr. Hans Kluge. Dr. Breda, we're looking forward to your presentation. Thank you for joining us today.
Thank you very much, Professor Mozialos, for it's an honor to be introduced by you. I am aware that I'm here between you and the official opening ceremony, so I'll try to be very brief. And of course, first of all, to express our gratitude for the invitation. This is a great forum. We've been here last year remotely, this year physically in presence. We're developing this collaboration with the university and with the medical school, which is really very important. WHO is very grateful for this collaboration and very happy and looking forward to establish a collaborating center in the future here. What I would like to do in these a few minutes, is maybe to bring us back to the beginning and maybe picking up a bit on what the Vice President Shinas really was alluding to in his presentation, which is bringing maybe a bit more of a perspective, which is public health oriented, and a perspective which is more inspired on the inequalities dimension and in the socioeconomic aspects. We've heard a lot about ethics, and we've heard a lot about individual approaches, the AI, the artificial intelligence, the machine learning, and how we can advance all these things on a, let's say, almost individual basis. But I think that at the same time, as we move forward in this, we really need to keep in mind that public health and the social aspects, socioeconomic aspects are also very, very important. This is why WHO and the UN really calls for a comprehensive approach which is really inspired by the Sustainable Development Goals, where health, SDG 3 is not seen in isolation, but it's working together with the other 16 Sustainable Development Goals. And here in this slide, a very simple illustration on how health should really come together with more responsible consumers and with the environment and with aspects related to, for example, food security, which is so important and it's becoming suddenly such a critical issue in the European region. So we really advocate to, as we move forward and we put our house in order, because we are talking about health and health systems, of course, and we need to take that responsibility. But at the same time, interacting and working together with the other sectors in a way that we address the socioeconomic and the social determinants of health is very important. Because particularly in the European region, we really have one very significant problem, which is the issue of inequalities. And if you look at this slide, I'm not going to go into details into it, but just to give an idea that the bottom 20% of the European region population experience more hardship in terms of quality of life, in terms of life satisfaction, in terms of mental health, in terms of their own self-reported health when compared with the upper quintile, if you like. So one of the biggest challenge we have as we move forward is how can we address these inequalities? First, do no harm. Many of these things that we see as innovative, based on technology, harnessing the power of digital requires that we do it in a very responsible way and that we address inequalities, leaving no one behind. And that is not always the case. That sometimes it's very difficult. And this is one of our preoccupations always in WHO, particularly in WHO Europe, as inequalities is really one of our main challenges. So sometimes we tend to be really medicalized, focus on what are the health conditions that affect more our populations, and we talk about cancer and we talk about cardiovascular diseases. And if you look at this slide from the global burden of disease, of course, these are by far the main causes of death. But these main causes of death that are non-communicable diseases most of the times in the European region, across the board, north, south, east, west, the fact is that they are linked with social determinants of health, and there is a lot of inequalities, which sometimes we are not able to address. And when we tackle it, sometimes we increase and we enlarge, we extend the gap instead of really closing this gap. And it's very important that when we address these main health conditions, we really make sure that we are doing it in a way that protect more the most vulnerable. And that is not always the case. So here in Greece, we have established a new office on quality of care, highly supported by the government of Greece. By the way, we are very grateful to the people of Greece for that. This Athens Quality of Care Office is really looking into supporting member states in the region, looking into innovations. And for the first time, we believe that we are trying to bring very strong focus in terms of the social determinants and the aspects of inequality into quality of care and quality of the healthcare that we provide to our populations. Why? Because that is very important. If you look at the main causes of death, again, in the European region, you would see that, for example, in terms of cardiovascular diseases, most of the issues we have, they can be attributable to poor quality of care. And this poor quality of care is probably more frequent in the most vulnerable groups. So I would like to argue that when it comes to really reducing inequalities across the region, we really need to pay more attention. Another issue, for example, we are not addressing properly, as simple as hypertension. Hypertension is one of the most simple, conditions that we could really address. You don't need sophisticated medicines, you don't need sophisticated treatments. However, we are not doing it properly. And we are not protecting particularly those who are more vulnerable. The older, the groups with lower income in the population. In this slide here, you see that around 40% of the population in most European countries, which have high blood pressure, they are not even aware And some who are aware they are not being treated. And some who are aware they are not being treated, and others are actually not being controlled. And only 1 out of 7 globally, and this is worse in many European countries, I mean, they're only 1 out of 7 are actually being controlled. So it's so easy. The number of lives you could save, particularly in poor groups of the population, is enormous. Not only in Greece, but many European countries, and we are actually not yet there. Therefore, I would argue that we have poor quality for the poor and for the most vulnerable. And we really need to go back to basics in this context. And when we are talking about innovation, when we are talking about particularly the digital innovations, we really need to make sure that we don't leave anyone behind and that we take that into account. Simple things like patient experiences. These are very important. Patients' experience reported by the most vulnerable are always worse across the board. And we are not doing enough in this context. This is very simple. Innovation is not only AI, machine learning, is also social innovation. And in terms of reducing inequalities, probably we are not doing enough. And very close to our heart, this is a very simple study. It's looking at young people adherence to the Mediterranean diet, the so-called Mediterranean diet. More than 50 systematic reviews showing that the ways of eating of Greece and Spain and maybe even Portugal and Italy and many Mediterranean countries could protect us. It doesn't do it anymore because we lost these traditional ways of eating. But even when it comes to adopting these traditional, let's say, behaviors, the most vulnerable are not there yet. And they are the ones that first lose those traditional habits. Therefore, I mean, wherever you go across the board, you will see that there is a clear difference between groups in the population, and probably we would need to do more and scale up our actions. We believe that in Greece we have a great opportunity. We have this new quality of care office, as I said, based in Athens. And one of the things that we really want to do, we want to address important issues like mental health. Therefore, we are initiating also a program for children and adolescents on mental health, because we believe these are vulnerable groups that need more support. And we are very grateful for the increase in our capacity, inspired by the European Program of Work, where really inequalities are at the center, addressing those inequalities, and again, making sure that we leave no one behind. In our vision for our quality of care office, I would like to say very proudly that inequalities word is written there. So it's about making sure that when you talk about quality, you need to make sure that you really address these issues and that you put the most vulnerable really at the center. So in a nutshell, I would like to express our gratitude for the opportunity today. We are very much looking forward to work with the Greek diaspora and the Greek colleagues in moving forward in terms of quality of care, but making sure that we protect the most vulnerable and that we put them at the center. Thank you very much.
Thank you very much, Dr. Breda, for this eloquent presentation and for raising the issue of inequalities. It was part of our program today just to talk about socioeconomic determinants, not only the ethical determinants. And it is great that you have outlined all the fundamentals that are quite important to consider in terms of reducing these inequalities. Perhaps next year, Professor Anastasiadis, we can have further discussions of the issues of the inequalities and socioeconomic determinants of health, going beyond the socioeconomic, I would say, because there are also commercial determinants of health, there are political determinants of health, policies and legislation enacted by the governments may have implications for our health, and the pure legal determinants of health, as we have seen recently by the more recent ruling of the American Supreme Court, that will have important implications for women's health. So the situation is far from being perfect if you look at global trends. So if I ask you, what is the country with the highest life expectancy in the world, most people will get back to me and say Japan. And obviously, amongst the largest countries in the world, Japan is the country with the highest life expectancy. But if we consider all countries in the world, even tiny countries in the world, the country with the highest life expectancy is the country with the highest concentration of billionaires in the world. And the country with the highest concentration of billionaires in the world is Monaco, where life expectancy is 89.5 years. Now, take that and compare this to life expectancy in the Central African Republic. Now, life expectancy in the Central African Republic is 53 years. Now, the life expectancy gap between the billionaires in Monaco and the very poor in the Central African Republic is 46.5 years. So people living in Monaco live 46.5 years more than those living in the Central African Republic. But you may say I'm terrorizing you by just comparing Monaco to poor Central African Republic. But if you look at New York City, the life expectancy between different neighborhoods is as much as 28 years. And this is the most recent data. Within the same city, you see differences up to 28 years. We don't have Greek data, unfortunately. It would have been the task of our National Statistical Service to provide us with this data. For example, it would be nice to know differences in life expectancy within Thessaloniki, for example, or within Athens, between areas where the higher socioeconomic groups are living and the lower socioeconomic groups are living. But that would be a task for our National Statistics Service to get its act together and provide us with more accurate data. Now, without further ado, I would like to thank my co-chair, Professor Anastasiadis. It's always a pleasure to co-chair sessions with you and have detailed discussions about the future of healthcare, going beyond the more technical and the more scientific that you're dealing with in your main program, taking a more phronetic perspective, I would say, the more Aristotelian phronetic perspective, which is going beyond episteme and techne, and it's about value judgments. It's about finding out what is the best for the people, after all. Thank you very much for inviting me here, and thank you to all speakers.
Thank you very much, Professor Moskalos. I think you have been already included as the organizer of this forum after these years that you are joining online, and I hope next year you will be able to be in person here on site, and we will co-moderate the sessions that you have already suggested to follow up and do another big event in these topics next year. So thank you all for your attendance, and hopefully we will see you down to the opening ceremony, which actually is in 2 minutes. Thank you, Professor Moskalos.